As good as yesterday, and also filled with many nice talks. We will start today with Professor Holm Ullich from the Translational Gastroenterology Unit of the University of Oxford, and he will talk about monogenic inflammatory bowel disease and genomics. Thank you very much.
Thank you so much. I think it's a brilliant meeting. With really many thanks to Anne and to Marielle for organizing these meetings that I thoroughly enjoy. So we talk about very early onset inflammatory bowel disease, which means 8:30 start. Inflammatory bowel disease is a very complex disorder, and the majority of cases are polygenic. And then there are a few outliers that are caused by a single gene defect that has a bigger impact. And here there is sort of this overlay with immune diseases, and that's why sort of the discussion and the crosstalk between gastroenterologists and immunologists is important. In a simplified way, the gut has to maintain a barrier function and immune homeostasis, and if any of these mechanisms fail, then intestinal inflammation can arise. Now, over the last 10 years, there has been a huge increase in the number of genes, and the important thing is to address this in a more systematic way. And Chrissy Bolton and Sumit Pandey and Chris Milley tried to do this and to establish a taxonomy, a systematic approach to monogenic inflammatory bowel disease. What they took into account was the penetrance of intestinal inflammation, or the expressivity, to take clinical phenotypes into account, expression of gene and proteins and biochemical pathway, and then to derive a model of a taxonomy. Now, if you do this, there's always the risk of overfitting, and so it was important not only to do this with a number of genes to derive this model, but then to validate it, and we did it with adding sort of new genes that had been sort of identified in the last 2 years, and by adding this we could show that this system with over 100 genes is stable. What it means and what the biologic meaning is that we have now an informed model where these monogenic IBD genes are expressed in terms of the epithelium, in terms of the mesenchymal cells. It's not all immune cells, but then similarly to this large number of genes that are dominantly expressed in phagocytes, in neutrophils in particular, or in macrophages and monocytes, but then sort of a large cluster that is partially overlapping and is expressed in lymphocytes as well. What this in the end suggests is that we have certain subgroups of genes that are expressed dominantly in phagocytes. So, for instance, key exemplar, this genes, for instance, chronic granulomatous disease that leads to intestinal inflammation in about 1/3 of the cases. But then certainly genes, for instance, that are involved in immune regulation, for instance, these IPEX or IPEX-like syndromes. Then there is this group of hypomorphic skids, whereas the selection defect is the likely cause of intestinal inflammation. And then you see there is at least 40% of genes that are not in these simple categories. They are overarching, they are expressed in multiple compartments, and this taxonomy now takes into account laboratory tests, stem cell transplantation, single cell expression, and these biochemical pathways. And this is one step to go into a more systematic approach, and the sheer number of genes suggests that we have to have systematic screening approaches as well. In order to do this, we derived some strategy how best to select these patients. Because IBD is a specific problem. If you look, for instance, for developmental disorders, the chance that you find a gene defect might be between 50 and 90%. if you go for immunodeficiencies, it might still be 30-40% that you're successful. If you take the overall number of patients with IBD, it's much lower. So we have to pre-select our patients. And one of the selection criteria here is age of onset. So if these patients are under 2 years of age, there's a very high chance of about 10 to 15% to identify a monogenic defect. And this sort of likelihood decreases with increasing age. So age stratification is one, and then we identified several subgroups that are important, and not surprisingly, infection susceptibility or autoimmunity as an indication of an inborn error of immunity sort of is important, but then there are some congenital intestinal signs, early onset malignancy, and then in addition, of course, a family history of a monogenic IBD, which is important. And by this sort of pathway, we can select patients according to their likelihood to have a defect. Now, if we want to translate this into clinical practice, then research paves the way. But then we need guidelines to address this in clinical practice. We need to have a healthcare system to take things on and allow us to do this on a national scale. And in the UK, the NHS has adopted this with a national genomic test directory that allows that all children who fulfill this criteria to undergo next generation sequencing. And then there is this important part of validation, revalidation, and research into treatments. Because what we want to do is not only to explain a disease, but we want to treat. This is only possible if we, and I mean we scientists, clinicians from different specialties work together. And so the multidisciplinary team approach is absolutely essential for the decision making, which patients should be investigated, for the variant interpretation, and then of course which care sort of these patients should undergo, what can we recommend to our patients. And I want to give you a few exemplars as snapshots showing the opportunities and the difficulties that we currently face. So, one exemplar. This is a patient from Oxford who was over 40 years of age, and I use this as an exemplar. I've shown sort of this patient before, but this patient is again like yesterday. There is a history, there is a story. This story does evolve. And what's happening is this patient developed intestinal inflammation with 11 years old, multiple operation, had a small bowel syndrome, over 25 operations, more than 1,300 days in hospital. When we identified a XIAP defect, this patient was already considered for a small bowel transplant. This would have gone wrong completely. So the patient now underwent an hematopoietic stem cell transplantation and subsequently a small bowel transplant. That's experimental medicine. What we need to do is, of course, to be sure that these defects are correct or genetic information. And we have now an increasing spectrum of tests that can be used, but many of those tests are still not in routine clinical care. They are at the edge of research. So for instance, for XIAP deficiency, the test with muramyl D peptides that signals via the RIPK2 and then allows the NF-kappa B signaling, and then we can either do functional tests, for instance, looking for antimicrobial activity, but this is a very cumbersome test. You can do it in a research lab. It's not in a clinical genetics or in a clinical immunology setting. In a clinical immunology setting, the FAX assay, for instance, performed by Kimberly, sort of to see the expression is useful for the majority of patients, not all. And the TNF induction assay can help you to see the NF-kappa B response. But again, it's not perfect. If you have a NOT2 homozygous variant, you would see exactly the same outcome. So you have to have a mixture of different tests. Another group of diseases very important are the IL-10 signaling defects. These patients develop with 3 to 6 months of age, and the test that most labs use is to see cytokine suppression by adding exogenous IL-10 to an LPS stimulation assay. Very simple. PBMCs stimulated with LPS, they produce buckets of cytokines, you add on IL-10 and it's suppressed. And if there is an IL-10 signaling defect, then this doesn't happen. And in this patient with an IL-10RA defect, we saw this absence of signaling. Now, not all tests are the same. We have a phosphoSTAT3 test, and this phosphoSTAT3 test did show that there is a functional defect, but it looked more like an hypomorphic variant. So again, we have to combine tests. One does typically not fit all. And before making decisions on hematopoietic stem cell transplantation, you want to be sure, because of course there is a risk. Then our knowledge base might be reduced and not sufficient. So we have seen in Oxford this patient who had about 10 years of inflammatory bowel disease, about 20 years of immunodeficiency with a congenital neutropenia. This patient had been seen in a center of excellence before. When we saw this patient with IBD, we expected all the neutropenia genes had been already investigated. So we did at this stage genome sequencing, and it turned out it was the most likely candidate. And this is another lesson. We cannot expect all these things. Sometimes we have to redo to reconsider. The other problem was that in this patient, very few had been transplanted before, but never with IBD. So we were not sure would it help these patients. The real problem was the inflammatory bowel disease in terms of quality of life. And the patient decided he wanted to undergo the transplant. He's happy. He's so happy he didn't come for follow-up tests, but he's supporting the community because he's even adding on to our guideline processes because he feels that changed his life. But there was no evidence available, and it's a big risk, and we face this situation all the time. Another situation, very difficult. These patients often have not only immune problems, but extra-intestinal and extra-immune manifestations. For instance, in human TGF-beta1 deficiency, where there is a brain abnormality there. And it's a huge ethical problem to consider whether a patient like this should be transplanted, which likely would correct some aspects of immunity, but certainly not all of the problems of the patients. And these are problems that should be addressed by a multidisciplinary team, and that's why it's important to discuss across disciplines. We yesterday discussed mosaicism. And mosaicism happens in inflammatory bowel disease as well, and this can lead to unusual phenotypes. So for instance, we identified a patient with an mosaicism in sub-B, no signs of infection, some folliculitis, inflammatory bowel disease, very early onset, and about 70% of the neutrophils, monocytes, are deficient of sub-B, and that explains 30% are still sufficient to keep these patients and prevent the infection susceptibility, but the 70% of neutrophils and macrophage who cannot properly digest bacteria, who produce inflammatory cytokines, probably drive the intestinal inflammation. And by identifying multiple cell types, we could predict that this defect must have happened at embryonic day 3 to 5, when the germ layers are just diverted. So we can really make predictions in time, and this explains the phenotype of the patient. We did not transplant, and the patient didn't want to be transplanted in this setting. This patient accepted that there is intestinal inflammation, difficult to treat, but he didn't want to undergo the risk of an transplant. Other causes, copy number variation. So in Paris, Nadine Serf-Bensosan has identified a defect with a duplication of the locus of the IL-2 receptor. This very early onset inflammatory bowel disease provided functional data. And it's very clear there is an immunological phenotype if you have too many copies of this receptor. We identified subsequently a patient with nearly identical phenotype. We validated the functional evidence, and that's really sort of reassuring. But is it the course of disease? Actually, we do not know, because if we look into databases of copy number, for instance in Decipher, we find sort of over 50 patients who have this mutation who do not have intestinal inflammation. And this shows that it's very convincing what we have on the data, on the phenotype, but we have opposing population genetics evidence, and it's probably more a modifier than a causative variant. Then often we start with a single patient. And Jean-Laurent Casanova has written a really sort of important sort of summary how to investigate single patients. And many sort of stories in immunology in monogenic IBD started with a single patient. So here we had a patient with an auto-inflammatory condition. Alexio Muis had identified this patient, and jointly with Ying Huang from Shanghai and many collaborators, including, for instance, Sophie, we investigated this patient, identified a gain-of-function variant in SYK, the spleen tyrosine kinase, and this stage we were stuck. We couldn't find another patient. Subsequently, multiple other patients were identified, but the causative information came from a mouse model. And this mouse model replicated aspects of the disease and allowed some prediction about the hematopoietic origin of the disease, which informed procedures, for instance, hematopoietic stem cell transplantation. So another kind of validation, adding models. But it's not all the immune system. And it's very important to keep in mind it might not be the immune cells that drive this disease. So here's a defect in goblet cells. So Aline, who's in the audience, is studying a disease, AGR2 deficiency, which leads to complete absence of goblet cells. The mucus layer is gone. There is increased translocation, and the inflammatory response is probably a physiologic response. You have more bacteria, the immune cells have to respond to this, and this is not an immune defect. So to study and to validate those defects, we need new tools that study the intestinal epithelium. And there is now a growing number of models, not only sort of the classical organoids that have been developed by Hans Clevers and others, but there are now microfluidic systems where you can add on multiple cell layers, where you start to see villus structures and crypts. And this is much more of a physiological setting. So let me summarize. We have more than 100 genes that are linked to monogenic forms of inflammatory bowel disease. See, more than half are linked to the immune system, but it's not all. For instance, some groups of immune defects are absent. For instance, interferonopathies do not present with intestinal inflammation. We have very good evidence that this is not a driver. So you cannot say, oh, immune system is sort of damaged, and this will lead inevitable. So there are certain groups that lead to intestinal inflammation. Then there are certain defects that are in compartments, and this can also include the endothelium, the nervous system, the epithelium. But then there are other defects that are cross compartments. And those are very, very difficult to judge whether, for instance, hematopoietic stem cell transplantation is curative or not. And the last point that I want to make, we have a huge gap between our genetic information and our treatment options. So hematopoietic stem cell transplantation is the strongest evidence for many diseases. For some it works, for others it doesn't. For instance, TTC7A sort of corrects the immune problem, but does not correct the intestinal phenotype. Gene therapy is coming up. There are certain exemplars that this probably is the future. There are multiple small molecules. There are biologics that are very, very promising. So, for instance, blocking IL-18 in XIAP deficiency or in some NLR defects is really interesting. Blocking IL-1 and IL-10 signaling deficiency. But in many of these cases, it's a single case report, or there are two patients. We wouldn't accept it in classical medicine as really good evidence, and we have to collaborate together to build up this evidence to allow our patients to make their best choices. With this, I'd like to thank our team. This is really sort of a fantastic collaboration within Oxford with many international collaborators throughout. And if there is somebody who's interested in the gut and wants to join, then please send me an email.
Thank you very much for this wonderful presentation. Are there any questions?
Just out of curiosity, I think I missed the gene for the neutropenia patient. I think you didn't mention which gene in the end you found. Maybe you could explain.
The spleen tyrosine kinase. SYK.
Oh, really? No, but anyway, okay. I come later and I ask.
Afterwards.
Yeah.
What's your threshold for going and testing genetics in patients that might get to us as adults? Would you still just watch at the age of onset, or that wouldn't be enough?
So you
like patients that come with a history of IBD, because a lot of the patients you showed are like in their 20s, 30s, and 40s. Would you test a person that in their 40s that's treated for IBD, for Crohn's, and get, I don't know, one of the patients who showed that pneumonia and was 40, I think it was the CEP one. I mean, would you test
so this is probably now sort of what we are facing. We have to work up, and there are still some patients around who sort of did not have the opportunity of genomics, who had sort of these criteria, who should be treated, who should not have the same history. We should be much, much faster now. And we still probably have quite a few patients that need a workup. The biggest sort of likelihood is the infantile onset intestinal inflammation. They should all receive whole exome or genome sequencing. In addition, the ones who are symptomatic and have extraintestinal manifestations if they are under 6. And in exceptions, even beyond 6, age of onset makes sense. However, irrespective sort of how old they are, or at least until a certain stage, if they had this history, these cases of this 40-year-old suggest it's not too late. There is a 40-year-old with 30 years of history, extreme sort of severe disease, XIAP deficiency, and this patient has now the best quality of life in 20 years. He really appreciates that despite sort of having sort of this experimental approach, and despite of taking medicines, his quality of life, being able to eat, being off parental nutrition, is for him the most important part. And that's what a point I wanted to make. The majority of patients are younger. The patients we see now are hopefully half a year old, a year old. We don't have this diagnostic delay, but it's important sort of not to forget that there are other patients around as well.
Just a question. The patient with CYBB mosaicism, did that patient have gingivitis and periodontitis?
Could you please repeat?
Did the patient with CYBB mosaicism, did that patient have gingivitis and periodontitis?
No, it was just folliculitis.
Okay, all right, thank you.
So there was some folliculitis that sort of was closest to zinc, did not even present with an, let's say, a female type of CYB. It wasn't sort of this classical sort of change. There wasn't sort of the butterfly sort of pattern. The folliculitis was the skin change that sort of was closest to the disease and potentially leading sort of to a suggestion that there is some error of immunity.
Thank you.
I also have a question related. Thank you very much. That was wonderful. Also on the somatic mutation that you've shown. So, and it's almost a bit of a philosophical question. This one was a very early embryonal mutation in terms of when it arose. One could imagine that there may be some mutation that just happen a little bit later. So then maybe onset would be a little bit later. Maybe the phenotype may be very locus specific. So, and I'm not in this field, but has anybody ever cared to get biopsies and sequence ultra deep, whether some of the more sporadic and later onset cases may even also have monogenic forms, but then in somatic state?
Absolutely. And this is sort of something we just need to understand, because in theory it would be possible that some areas of the epithelium do have sort of a region of half a meter, and that might explain why these patients sort of have flare-ups, are difficult to treat. They might have a Nemo variant in the epithelium that is driving this, and it might require sort of to take biopsies from the part and then to do a systematic landscaping, a cartography of the of the gut. Absolutely, but it could be that this is a layer down in the fibroblasts, and the fibroblasts just don't make sort of the basal layer, and that's why the epithelium is affected. But absolutely,
but nobody started doing that. I mean, the sequencing itself is getting trivial. So
it is not trivial, because if you take the biopsy at the wrong place,
sure, no, that's what I'm saying. The sequencing is trivial, but getting the right material is not. So and you would be in a perfect space to organize that.
So we currently try to see what single cell sequencing sort of the real sequencing would be the way to go, learning from the oncologists there, and then to do this in a cell segmented pattern. There must be something, absolutely. I think it's sort of there is a lot to discover, must be.
Yeah, agree.
Okay. Well, thank you very much, Holm. And then we continue with several challenging cases from expert centers. And the first one we start with is from Sophie Hamilton.
Okay, thank you. Good morning. It's hard to follow that, Holm. But I've brought two cases that taxed us in Newcastle for you to have a think about and to chew over. So I want to take you back to 2014. I can't remember how many genes we were at then for primary immunodeficiency, but it was quite a lot fewer than we have now. And we were presented with a 1-year-old child who was the first child of parents who were first cousins. And she'd been absolutely fine when she was born, but around 6 months of age, she came to see the general paediatricians at my hospital because she was having some loose stools, sometimes with blood in them, and it was thought that she might have cow's milk protein intolerance. But she was quite well grown at that point, and nobody was too worried. And then the family went off to to visit relatives in Pakistan for a prolonged period. And while she was there, she got really quite sick with more diarrhea, significant weight loss, and she was also feverish and came home in a bit of a state. So she was admitted under the care of the general paediatricians, and they told us about her. So we went to have a look, and she looked terrible. She was very wasted, apathetic, lying on the bed, not interested in anything. And on her basic bloods, she was anemic, her albumin was down. And there was no obvious infective cause that was driving her diarrhea. So we suggested some basic tests, and some of these showed that she had a CMV viremia, and she also had some gut biopsies on endoscopy, which showed some inflammation, and CMV was detected on PCR, although we didn't see any, megaloblastic cells. So let me show you her peripheral blood immunophenotype. I'll just give you a little moment to have a look at that. So these are cells per microliter on the left, and you can see she had plenty of T cells apparently, but if we look in detail there, they were mostly CD8s with an abnormally low proportion of CD4s, and none of them looked naive, so it was a really abnormal T cell compartment. She was making IgG, and she had some vaccine responses, but no pneumococcal antibodies, and her IgA and IgM were a little bit low. So obviously we asked ourselves what type of disorder was present, and cogitated about how we were going to look after her. So I put those questions to the audience.
So because also the audience on site, if you have answers, please take the microphone so the online participants can participate as well, can hear the question.
I mean, if we think in terms of IUIS categories, for example, what kind of disorder do you think this might be?
So a combined immunodeficiency, maybe MH class 2 or something like that deficiency. And further investigation, I will perform TREC and T proliferation test and functional test to deepen.
Perfect. Yeah. So, it's the first question of the day, so I made it an easy one in terms of what kind of disorder. Yeah, of course, it seems as if it's a combined immunodeficiency, and indeed, here are the proliferative responses that you asked for, and she had very severely diminished proliferative responses to PHA, but interestingly, relatively preserved responses to some other stimuli that bypass the T cell receptor. So it looked like some kind of T cell activation defect, and reassured us that this wasn't some kind of weird, it wasn't a DNA repair problem, it wasn't a funny kind of omens or anything like that. It looked as if it was a real T cell problem. So we managed her as a profound combined immunodeficiency. We put her in protective isolation, we treated the CMV, we prophylaxed her against other sorts of pathogens, and we gave her nutritional support. And of course, we did some molecular genetics, which were a bit limited through the health service at that time, so we went down a research route, and we worked her up for stem cell transplant. So the diagnostic workup that she had was whole exome sequencing, and we screened the data against the contemporary panel of IUIS genes, and we also did a kind of gene agnostic, open the exome type look at other genes, screening for rare, non-synonymous, predicted deleterious variants with an autosomal recessive kind of model, and we didn't find anything. But nonetheless, she clearly had something seriously wrong. So we went ahead with a stem cell transplant using a slightly mismatched unrelated donor. And she did really brilliantly. had fantastic immune reconstitution and is basically a normal child. But for her family, this is still a big unknown. The assumption was, because parents were related to each other, that obviously there was a high likelihood that this was an autosomal recessive disorder. But they were thinking about whether to have some more children, and we thought we should have another look. So we looked back at the original data, and then we thought, you know what, let's do whole genome sequencing. So we did that, and you keep tweaking your pipeline. So our pipeline for whole genome sequencing makes us look at all rare variants. And she turned out to have a homozygous synonymous variant in DOC2, which you'll be familiar with as a disease gene relevant to this presentation. So it was rare, not in population databases, and I'll talk in more detail about the predictions, but it was predicted to be damaging. So here's the original report that came out the year after this patient had presented to us, and the distribution of loss of function alleles through the predicted protein structure, and I've highlighted our patient's variant, which was at valine 606. I'm not going to talk in a lot of detail about DOC2, but it's basically a sort of adapter downstream of a lot of stimulatory cell surface receptors and activates RAC, kind of connecting to the cytoskeleton of many leukocytes, not just T cells. And in that original report, sorry, that's a bit small, but the descriptions of the patients are remarkably similar to the child that I've outlined to you. So reduced T cell proliferation, CD4 lymphopenia, viral infection, chronic diarrhea. Tick, tick, tick. So both mutation taster and this slightly newer tool called Splice AI predicted that this would be a deleterious mutation by introducing a splice site in the middle of an exon. So I've highlighted there the wild type splice site. Basically, the mutation creates a new one, or at least that was the prediction. So how would you test that?
At RNA level?
Yeah. So what sample would you do that on?
I don't know where it is expressed.
Yeah. So this is a child who's had a stem cell transplant and is 100% donor. So this potentially presented a challenge to us. Although I suppose we could have gone to parents who were heterozygous for this variant, and we could have had a look there. As it happened, we had access to both dermal fibroblasts and we had one aliquot of frozen lymphoblasts from the patient. So we checked the literature, and it turns out that this is a gene that is transcribed in a wide variety of cells, including fibroblasts, although they don't express the protein. So we did the RNA sequencing in the patient's fibroblasts and confirmed that all of the message was spliced using this cryptic splice site, which messes up the sequence and results in a frame shift and a truncation. And then with that last vial of lymphocytes from the patient, we did a Western blot to show that there was no protein being expressed either. So that was quite satisfying, because all these years later, we were able to make a diagnosis that completely fitted with her presentation and now enables this family to screen future pregnancies. So we were pretty pleased about that one. Especially, I used to use this case to demonstrate how annoying it was when we couldn't make diagnoses, and now we can. Have I got time for another case, or do you want me to just stop?
Let me see. Actually, yeah. Officially, it's up to Gigula now, next center. But we can. Is it okay? Yeah. So now you have to stop, so we can
I'll
go
stop.
to the next, and maybe afterwards we feel sorry. So,
I'll just give you my take-home messages.
We skip the coffee, so so the audience wants to skip the coffee. So yes, keep on.
I'll make it shorter. Okay, these were the take-homes there. So remember synonymous variants. We can look at their effects using nice fancy tools, and remember to save material from your patients. Here's another one, just quickly. So we had two siblings, both of whom presented very early in life with horrible HLH, hemophagocytic lymphohistiocytosis. The first child died before transplant even. The second child, we couldn't really screen, and he was already symptomatic within about 2 weeks of birth. Luckily, we were able to transplant him. And we knew from the first time around that there was a single pathogenic coding variant in STXBP2, which is a known gene for this phenotype, that was paternally inherited. But with whole exome sequencing, we hadn't been able to locate a second variant. So, you know, the question is, has this got anything to do with what's going on in this family? We hypothesized that, of course, there was a second variant that we hadn't been able to pick up somehow, presumably because it was in a regulatory region or extended splice site, or perhaps there was some structural variant that was disabling one of the maternal alleles. So I'm going to answer my own questions. So we performed whole genome sequencing here to look for that. And satisfyingly, we did find a very rare maternal variant that both of the children had inherited. So this was potentially consistent with compound heterozygous inheritance, if you like. So it's very rare, and it was a little deletion in intron 5/6 that was one of the few rather conserved regions of that intron. So our suspicion that it was relevant increased. And so to test that, we performed a PCR that was anchored well either side of this on cDNA to look at what the splicing was like in cells from this family. So usually, if you do that in a healthy control or in the father, you just get a single band. But when we looked at the patients and their mum, there were two bands. So that was consistent with a splicing defect that had been maternally inherited. And when that gel on the left is a better separation. There were actually two abnormal products on the upper band, both of which had retained intron 5/6, and in one there was also skipping of exon 5. So the consequences of that were that, in fact, there was nonsense-mediated decay and very little message in the patient cells, nor did they express the protein, and that explained the failure of NK degranulation. So sorry, that one was a little bit rushed, but just to emphasize that if you have a single variant in a gene that is a very good fit for a severe phenotype, it's really worth pursuing the missing second variant. And again, to emphasize that being able to validate these sorts of novel variants is really important to prove that they are causative. I'll stop there. Thank you very much.
Well, thank you very much.
Questions or coffee?
So our next presenter is Giuliana Di Matteo, and she's from Bambino Gesù Hospital in Rome.
So, good morning to everyone, and thanks for the opportunity to discuss this issue. So, the aim of this work was to investigate radiosensitivity in patients affected by RC1D deficiency and suggest this radiosensitivity as a new trait in this disease. This study started from the characterization of a ESID patient with immune dysregulation affected by gastrointestinal bleeding, perianal and oral ulcers, anemia, and this since the first years of life. Now this girl is 22 years old and born from consanguineous parents. Over time, she continued to have a severe picture characterized by several recurrent infections, IBD, psoriatic dermatitis, autoantibodies, hyper-IgA, hyper-IgE, but only recently she developed a T-cell lymphopenia. Interestingly, these patients showed an increased ionizing radiation-induced sensitivity. That means that irradiation-induced DNA damage, and this damage persists in the cell of the this patient. This increased radiosensitivity is found in fibroblast. Non so se, ecco, okay. In fibroblasts of these patients as an increment of H2AX foci in fibroblast after irradiation, and also in EBV cell lines as an accumulation of chromatide aberration in our patient, and non si vede, vabbè, okay. In our patients, although at low level compared to other non-radiosensitive disease as ataxia telangiectasia and Nijmegen syndrome. Molecular investigations were started at different time, and after a first whole exome sequencing.
Would you like to use the pointer, no?
Yeah. Ah, yes. I'm sorry, I forgot it. So we performed several molecular investigations, including Sanger of different genes. First West that revealed an homozygous mutation in the SLC6A19 gene that is responsible for Hartnup disease. That is a metabolic disease that it's present with a defective amino acid transportation, and is characterized by the increased tryptophan in urine and severe dermatitis. But in our opinion, this gene couldn't explain the severity of this picture. And so we continue with the second OS that didn't reveal any mutation. And finally, EUGS revealed the presence of a deletion in RC1B gene. RC1B deficiency is characterized by all the clinical manifestations that I showed before. It's characterized also by thrombocytopenia, usually, but in our patient, this phenotype was low represented. However, we obviously confirmed the lack of RC1B protein expression, and also a reduced T-cell migration that is already described in previous RC1B cases. We also confirmed radiosensitivity in our patient, in our index case, and in other two RC1B patients in fibroblast and also again in EBV cell lines, although at different level. Then we change our strategy using a more reliable radiomimetic reagent to avoid the use of ionizing radiation because it was unavailable in our lab. And we started this new strategy by using bleomycin in EBV cells and to analyze not only the increment of H2AX positive cells after treatment, but also we use the immunofluorescence to confirm the increment of H2AX posi in EBV cells. This is the data of immunofluorescence. Again, after 2 hours of treatment, and in this case 4 hours, because in this case it was an optimal time to see this aspect. We found in all tested patients an increment with a certain variability between samples, and then we see we see this. This accumulation of H2AX positive cells, again in EBV cells, and importantly, this H2AX accumulation was particularly evident in G2M phase of cell cycle, and as shown here. We extended the time points of this observation, finding that even after 24 hours remain some of this remain this damage, this defect. But what's the role of RC1B? RC1B is involved in as a dual role. The first one in the context of R2-3 complex that is responsible of the formation of actin branched network, and it's a network showing this particular structure, Y structure, responsible to generate forces responsible for many cellular processes as survival, proliferation, mortality, secretion, signaling, and DNA damage response. And recently, in 2019, is also shown that R2-3 is an important complex responsible for drive the clustering of double-strand breaks induced by, for example, ionizing radiation. To cluster this defect, this damage, close to the complex responsible for homologous repair, specifically for homologous repair. Indeed, the homology-directed repair require a large chromatin movement respect on the non-homologous repair. And R2-3 is an important complex that do this, that drive this clustering. Another role of RC1B is known from many years, and in this case, RC1B acts as a chromosomal protein that is a substrate and activator of RRA that is responsible to determine the progression into mitosis and determine the progression of cell cycle. So, this data seems to suggest that RC1B is responsible of probably a defective R2-3 complex in homologous repair in G2M phase, but also independently an impaired RC1B role could determine the lack of activation of RRA and so the progression into cell cycle. So what do you think could be the impact of a similar defect in this patient? What do you expect to be the consequence of a similar damage in homologous repair or the persistence of a damage in our cells. What could determine? You can also.
Well, it may explain why she progressively is getting worse, but I think the real concern would be malignancy of some description with a repair defect.
Yes, this is one of the possibility. Some other suggestions? So actually, we don't know in this case. No, we don't know because the possibilities are different. So a defective RC1b determine a defective DNA damage response seems to suggest this, our data seems to suggest this. But a defective DNA damage response could imply, obviously, a misrepaired double-strand breaks, and so inducing transformation and malignancies, but also in the case of the double-strand breaks are not repaired, probably a precoce cell death of these cells, and so the consequences, for example, lymphopenia, or a tolerance of the DNA damage. In this case, probably the cell cycle, the proliferation of cells lead to a precoce senescence of these cells. So, in any case, to a dysfunction of some subset of cells or other. And there is also a defective cell cycle progression that we didn't focus. Many questions remain unanswered, but probably a combination of all these consequences could induce so the clinical relevance remains to be investigated. Future perspective could be to monitor the disease evolution in terms of what I have said, and we are using a lentiviral vector to confirm that this increased radiosensitivity could be restored. And we would like to explore, and I suggest to explore this trait in other actin-related primary immunodeficiency, and to explore also the differences between an impaired non-homologous repair respect to an homologous repair, because this mechanism could have a very different impact. It's not an high radiosensitivity could correspond to malignancies, but a low radiosensitivity is not necessary. Could have the same impact as showed, for example, for BRCA2 that is involved in homologous repair. So thanks to all. And these are the person that helped us to characterize this patient. Okay.
Thank you. Yeah.
How sure are you that also non-homologous end joining is involved or defective?
Sorry?
The non-homologous end joining pathway, did you hypothesize it's defective, or you have already done direct studies for that?
Yes, we think that the mechanism, that it seems that the mechanism is that of homologous repair. No, this is the question.
Now, during your talk, you talked that homologous recombination is defective, but in your last sentence, you said also non-homologous end joining.
Yes, no. The problem of that, I couldn't show everything, but we used, in comparison, Wiskott-Aldrich patient samples. And in this case, it's not clear. Probably Wiskott-Aldrich cells used prevalently a non-homologous repair, but they are part of the same complex, R2-3 complex. So probably they could have a different rule, and they use different repair mechanism. Although both show a defective repair. Also in viscoautogenous patients, we could, some has already known that there is a defective DNA repair. But this repair is independent on the phase of the cell cycle. Indeed, in this case, seems to be very specific. The rest in G2M phase is a peculiarity of a C1b patient. And this also could be a protective, maybe arrest in this patient. We don't know.
So there is one question from the chat from Sarah Signa. So since few patients with RRPC1B were transplanted, should the conditioning regimen for this patient be evaluated in the light of your results?
Yes, obviously. I think that we could better. It's important to confirm this trait because at this moment we have used EBV cells that present an high proliferative rate. And so we have seen a large variability in our samples. We use many controls and repeat many times, although chromatide aberration is a certain data. But in the case of a confirmation, it could be important to suggest a specific treatment for these patients.
So our next speaker is Kimberly Gilmour from London.
First, I'd like to thank the organizers for having me. I'm also going to present a couple of challenging cases. Hopefully this will be somewhat interactive and somewhat of a review for some of you. So just to remind you what we're here and why functional validation is important is that we can do some things. We can do in silico predictions, but that's best computer modeling. It doesn't mean it's actually going to be pathogenic. So then we can do a variety of things that's already been elegantly shown by the speakers already in terms of protein expression, in terms of protein function, and then looking at the whole pathway. As Sophie emphasized, and it's incredibly important, you actually need tissues and cells to do this. And they need to be the right tissues and cells because the proteins may not be expressed in all tissues. So case one is a very old case, but I think there's a huge amount of learning like Sophie's to be had from this case. So Granddad, I first knew Granddad in his 60s. He'd presented much earlier on. He'd presented in the 1980s to David Webster, who's a very old name for some of you. And he was one of the first patients in the United Kingdom to get replacement immunoglobulin. And back in the 80s, it was intramuscular immunoglobulin. So he'd been around for a while. And he was an atypical CVID. He had absolutely no NK cells. T cells were a bit low. He'd had a very long history of disease and was even in the 80s a late diagnosis. And he had his immunoglobulin and prophylactic antibiotics, and people were trying to keep him. And he was reasonably well, and he got married. He had a daughter, and that daughter had a grandson. And that grandson, at the age of 5, started looking like his grandfather. And Mum said, well, he's been sick. He's had a lot of infections, little trips to the GP, came to an immunology center, and had no NKs and low T cells and abnormal immunoglobulins. So what would you do next? Must be XLA, okay? Clearly, I think an excellent condition is definitely what we're looking at here. So what did we do next? Well, we did do immunophenotyping. What I haven't told you is that the B cells were in fact normal, and it was the no T cells, no NK cells, and low T cells that were the issue. So because of that phenotype, we looked at gamma chain expression. And on the top, we have, how do I get my little magic pointer? Sorry. Here. Excellent. So up here we have the controls. This is an isotype control and just surface expression of gamma chain, which you can see is quite normal in the control. And here's grandson, who was clearly abnormal, and granddad, who is also abnormal. And they were abnormal in a very similar pattern. So that was very suggestive. It was gamma chain SCID. We also did STAT5 phosphorylation. We could get more blood from the grandson. We were seeing he was being seen more regularly. So this is STAT5 phosphorylation to IL-2 and 15, unstimulated stimulation of 2 and 15. You can see the grandson really doesn't have stimulation. So we have abnormal gamma chain, absent STAT5 phosphorylation. What would you all do? You'd sequence the gamma chain. I mean, there's a lot of evidence it's gamma chain SCID. You have an X-linked disease, you have a phenotype, you have no abnormal gamma chain, no STAT5, you sequence gamma chain SCID. Exactly what we did. We just sangered gamma chain, didn't find anything. We then did a targeted panel, and it was our Tiger panel, and we did that because it had very high coverage. We were getting 300-fold coverage of the gamma chain. So we thought maybe there's something funny going on, maybe there's something we're just not picking up by Sanger, maybe it's in a different gene and we're just barking up the wrong tree. Didn't find anything. So then we put whole exome sequencing because clearly that was going to help. That didn't find anything either. We also included all the exon-intron boundaries in our sequencing, and at this point we'd learned the hard way. We'd also included the poly-A tail because we had a previous patient who had a very similar phenotype in the no gamma chain, no STAT5, in that case no cDNA, that we actually did gene therapy on, and it was only about 10 weeks after he'd had his gene therapy, we finally found the mutation in the poly-A site, which fortunately was gamma chain, and that patient is alive and well 20-odd years, not quite 20, 17 years post-gene therapy. But we looked hard for that. So we looked for everything we could. And then we did a little more. So the daughter-mother, so the daughter of the grandfather, the mother of the child, had non-random exon activation or T cells, which is highly supportive of gamma chain SCID, and we could show the grandson had inherited the inactive X. So clearly, this was gamma chain SCID. So what would you do next? Yeah, home? Promoter region, exactly. Whole genome sequencing is the only thing we haven't done. So we did do whole genome sequencing. And what we found was this mutation in the ETS binding domain. It was a single nucleotide change. And was that the underlying cause? It fit very nicely. It looked very nicely. But the gamma chain promoter has only really been well characterized in vitro and in mice. You don't know that this is really what's happening. Mice with gamma chain don't look quite the same as humans anyway. So was this disease causing, and how could we prove it? You could do RNA. RNA wouldn't necessarily help you, though, because RNA, in fact, we did do RNA well before this. It was low, but it doesn't tell you that that's the mutation. It just tells you that you have something that's causing very low RNA. Pardon? Yeah. You could test, yeah, that's exactly what we did. So we did it a little more complicated than we probably needed to. We actually made a lentiviral construct and transduced cells and did an old-fashioned complementation assay where you put back the missing gene and do you restore function. And that's exactly what we did. So up here we have gamma chain. So we have empty the cells without any vector, the cells with a vector that didn't contain the promoter, the cells with a vector that did contain the promoter, which restored gamma chain expression and also restored STAT5 phosphorylation. So by doing the functional complementation assays, I think we have definitive proof that that ETS binding domain was the underlying disease-causing mutation. Okay, so that's a quick, very simple story of gamma chain, but you knew what it was and you couldn't prove it. And actually, it's really important to prove it because it impacts family counseling, it impacts therapy. We're not supposed to do gene therapy if we don't have a mutation. That's part of the guidelines in the UK. So it is important to actually prove that that is the definitive mutation. I'm going to go on to sort of a very different story. And these are two siblings that presented with severe neurological deterioration and long mitochondria after a viral infection. So a bit different than what we used to look at in immunology, at least. So they're siblings. They were normal until 12 months of age. and they both got severe neurological disorders after the MMR vaccination. And the presentation was suggestive of mitochondria disease. Sorry, I'll click through these. But because you may or may not be mitochondrial experts, I certainly am not. I just wanted to remind you that the mitochondria do all kinds of things that regulate all kinds of parts of the body. There's a series of defined genes that have been well defined. This is what normal mitochondria look like. And importantly for this talk, the mitochondria go through this cycle of fissioning and fusing. So the mitochondria fuse to make big mitochondria, and they fission, and that gives you more mitochondria, and they fuse, and they go round and round the cycle, and now and then they get degraded, and some new mitochondria are made. But that cycle is important. It's what all of our cells are doing all the time. So we felt these children might have a mitochondrial disease, so we looked at the classic parts of the mitochondria. The respiratory enzyme chain activity was normal. By light microscopy, they were normal, but by electron microscopy, they were abnormal. And what they looked like is they had very long mitochondria. I don't know if you can see these. These are just massive. And if you look at the control, see the control has these nice little rounded up normal mitochondria. These have these massively fused mitochondria. And you can see it in muscle, you could see it in fibroblasts, and when we went back to do confocal, you could see these very long fibroblasts, long mitochondria versus the normal little ones. But it was most apparent on the EM that you just have these massive mitochondria. So what was causing it, and what do we do? So you have long mitochondria, happens after MMR vaccination. What do? We all sequence. In fact, we didn't sequence a whole genome to start with or a whole exome. We sequenced the known genes that we knew affected fission because there were only it was a limited gene set. So basically they went on a little mitochondria panel, had a few more genes than this. Did we find anything? No. So then we went on and we did whole exome sequencing. And as you all know, we looked at all variants, rare variants, anything that was deleterious, put on a mitochondrial filter, didn't find anything. Looked for things that might be compound heterozygotes shared between only the siblings, and didn't find anything. Looked for homozygous variants shared between the siblings only, and we found one gene. Happened to be STAT2, which had been very nicely described by Sophie and her team previously in patients with severe viral infections, just to remind you all that STAT2 is part of the interferon alpha signaling pathway. So what would you do next? For function, what's the easiest function to do? What's the easiest thing to do that we've already seen? Yeah, look to see, are we expressing this protein? Is this protein even being made? Because if it's not being made, then you have a lot of evidence that that might do it. We can do some in silico as well. So just to show you that we did do the sequencing, and here you can see the parents are heterozygous. Here you can see the affected sibling. Here's the healthy control. This is just showing the two children. This shows you STAT2 and where this mutation is sitting, and it's sitting right in an SH2 site, so a very conserved region, and this is conserved all the way down to fruit flies. So in silico, it was predicted damaging because that is very high conservation for a change. And then obviously we did the Western, and here you can see the healthy donor, a healthy control. That in fact was dad and the two children who clearly don't express STAT2. And then because we had Sophie's patient who had been previously reported who also had a mutation in STAT2, we could then look at their fibroblasts as well for mitochondria. And here you can see patient 1, 2, and patient 3 also has these very long mitochondria. So we have 3 patients with a fission defect with a mutation in the immune pathway. Because STAT2 is an immune defect. It's interferon alpha. It causes viral infection. We all know that. Why is it doing things for the mitochondria? So we actually felt we had to go on and prove that STAT2 was causing this mitochondrial abnormality. So again, we made lentiviral constructs and we did transduction experiments, and what you basically can see here are the patients And the patients, once they're transduced, and you can see they go from having long mitochondria to much shorter mitochondria, and that's very comparable to healthy control. And interesting, looking at some other mutations, STAT1 patients have small mitochondria. This is specific to STAT2. So what was our hypothesis? Because there has to be a reason that STAT2 is doing this. We have evidence that STAT2 is doing it, but why? Well, in a normal human being, you have this balance between these two serines being phosphorylated, serine 616 and serine 637. And we hypothesized that the phosphorylation, rather than being on 616, was on 637, which was pushing the cells towards a fusion defect rather than a fission defect. So we felt the balance might be off in that. So we did a series of phospho experiments. So here you can see the patients. Here you can see DRP1, which is present in all the patients, but you can see they have limited serine 616 phosphorylation and lots of serine 637 phosphorylation, and that's driving them to this fused state. Okay, and therefore we were able to prove that our hypothesis is that STAT2 interacts, and we've done some co-IPs and things, with DRP1 and is driving phosphorylation to the wrong serine. If you don't have STAT2, you phosphorylate the wrong serine, basically. There's still some work going on to do some more clarification with that. But I just thought it was a nice story of an immune gene, and we had a lot of I had a lot of assumptions. I did my PhD many years ago on STAT1, 2, and 5, that this was an immune system gene, and then it turns out that it's affecting mitochondria, and then having to show that it really is the STAT2 affecting the mitochondria, and we don't have two different diseases going on here, I think was really important. Just to remind you, when we do whole genome sequencing, we're getting a lot of variants of unknown significance. We can use Sanger, or in the cases of deletions, things like MPLA to confirm them, but functional validation is really important. I would argue that we're all doing these functional assays, and when it's a really nice story, you publish. But often, at least in our lab, we're not publishing; we're just confirming. So, you know, this is a new variant in, you know, CYBB. Can you just do a DHR, GP91 expression, tick done abnormal? But no one's described that particular variant before, and now we know it's pathogenic. What are we doing with all that data? And it's happening for gamma chain and stats and whatever your favorite molecules are. So I think there is something really important about sharing the data, because if it's the first time or it's a really interesting case, you publish it. But how many of us are sitting on a lot of functional data that's completely unpublished, has made a difference for the patients, but we're not sharing it as a community? And I think there's something we need to think about and do with that, so that we're not all reinventing the wheel. We do not all need to spend our lives doing stat phosphorylation DHRs if someone's already done it and shown that it's normal or abnormal. That was just my plea. I need to acknowledge lots of people for doing this. So there's a whole team behind the STAT2 work and the ETS binding domain. Fang Zhang did all the complementation assays, and Sergey did the sequencing for that. I have an amazing lab with some amazing staff who just now, it's a clinical lab. It's ISO accredited. Most of the tests we're offering are now ISO accredited, but they're great at, okay, how can we do that? And for those of you who work in a clinical lab, that's been a change, because actually when I first started it was, oh no, we can't do that, and now they're wonderful, and it's how can we do that and make that work? So I just want to acknowledge all of them. Thank you, and I'm happy to take any questions.
Thank you, Kimberly. It was a really nice presentation, and especially your last comment, that all the variants and functional data should be reported somehow. Do you have an idea how that could be done?
I mean, I don't know if we can put something either, and if people would actually use it. So years ago, we had all the little PID bases that were being run out of Finland, and actually were great, and they've sort of, I think they still exist, but we're not really good about putting not only the functional data, but let's be honest, my lab and we're not great about even putting in our mutations into databases as much as we should be. So I think there is something to be had, whether it becomes, I mean, there are mutations now in the ESID database, whether there's a field for functional validation for VUSs, whether it needs to be something different, whether we need to work the way we did with the HPO terms and do something very, I think it needs to be very international, whatever it is. but I think it's something actually that ESID really needs to lead on, because I think we're the largest group of immunodeficiencies, or ESID, CIS, to work together. Yeah.
Yeah, just to comment on the latter point, it's not exactly on what you emphasize here, but we are working on behalf of SolveRD. We now have established a framework that is called RDMM Europe. So if you just go to the SolveRD site, you'll see it. And this stands for Rare Disease Models and Mechanisms. And we are connected with the Canadians, the Australians, and the Japanese doing something very similar. And that is in essence matchmaking. So I think a lot of people now know matchmaking exchange, where clinicians and geneticists meet and they find mutations in the same gene. This is also including functional laboratories to matchmake them to respective clinicians and scientists. So everybody should be encouraged, if you have a certain model system or specific expertise on a rare disease gene, put in your name, put in your details, and you may be matched to other groups globally via this RDMM consortium.
Yeah, I think that's a really good point, and we all need to do that, but I do think we need to come up with a way of banking all of this data.
Yeah. Within ERN RITA Molecular Testing Working Party, Kimberly, there is a list of all the functional tests offered diagnostically in the different expertise labs. So the diagnostic test you would already be able to find, but not the data indeed.
So you can find out who does the test. It's just finding out the results and the gene variants together, which would be really nice. Thank you.
Yeah, thank you, Kimberly.
So our next speaker is Vanessa Sancho-Shimizu from Imperial College in London.
And our next presentation will lead us from clinical cases to healthy newborns. And also there are clinical cases. So Mirjam van den Burg from the LUMC, the Netherlands, will talk about challenging cases from neonatal screening of SCID.
The first challenge is finding my... no. I don't see it. Oh, here, yeah. I'll find it here. Thank you very much. Thank you, Marielle and Anne, for organizing this meeting and also for giving us the opportunity to have these discussions, and also that you start thinking about other things. And I think it's really inspiring. I was asked to present a challenging case for newborn screening, but I think newborn screening itself is very challenging, especially if we are thinking about new steps we would like to make. And I will start with this slide. Diagnostics is not screening. This might sound very trivial, but when working with screening and coming from a background from diagnostic, it's really a different field of screening. And if you see what the differences are, put them together, then first of all, the purpose is really different from screening and diagnostics. For screening, you aim to detect diseases or risk factors for disease in early phase. And for diagnostics, you want to establish the presence or the absence of the disease. And the target population is, of course, different because here the screening is more public screening. And for diagnostic, you have a patient with symptoms. And the test methods is also different because the test method for screening should be simple and acceptable to the general population. And for diagnostics, especially the academic diagnostics, what we also talk about during these meetings, then it's much more extensive. So therefore also the costs are different, and also the interpretation of positive results are different, and especially the thresholds for screening should be such a way that it's chosen towards a high sensitivity, and for diagnostics rather towards high specificity. If you think about newborn screening and for inclusion of conditions for the screening program, then we always go back to the criteria from Wilson and Junger. They are already defined in 1968. And here there's a whole list. I would not go through the whole list, but the condition should be an important health problem, and there should be an accepted treatment for patients with the recognized disease. and it should also be the treatment should also be possible to be given, and the patient also need to be followed up. Now, when we think about inborn errors of immunity, then we have the first screening for SCID. And that's already, I think, known for all of you, but I briefly go through it. It's based on the quantification of the tracks. And tracks are formed during the recombination of the T-cell receptor alpha locus. For a T-cell to become alpha-beta positive, you need to delete the delta locus, which is in between. And this recombination, most of the time via the recombination of delta that can pseudo alpha, results in the T-cell receptor excision circle with a circular DNA product which can be quantified by PCR in blood, but also on dry blood spots taken after birth. Now, for the SCID screening, then the US was really ahead. They started the pilot studies first and implemented, I think, now the whole country. And this is a slide I took from the PIT Live Index from the IPOPI website. And you can see which countries are already screening or are doing pilot studies or are considering pilot studies. I give you the results from the Netherlands, but it's not important from which country, because I think every screening program gives more or less the same results. So in this study that was called our SONET study, that was from April 2018 until December 2020, and we screened 200,000 newborns. And of these 200,000 newborns, 62 children were referred. And of these children, only one had SCID. There were a couple of other diagnoses, cartilage hair hypoplasia, Rothmund-Thompson syndrome, and also T-cell impairment syndromes, and also secondary T-cell impairment, idiopathic lymphocytopenia, and also false positives. So it's a whole spectrum of diseases, and the target disease is SCID, or severe T-cell lymphopenia. So this also thinks that it's maybe not specific enough. And there's a challenge for the SCID screening to improve it, because it's good to have, you need to identify the SCID and also the T-cell deficiencies who required treatment, but there's also non-actionable secondary findings, and especially the false positives you would like to prevent. So there are different options to prevent the non-actionable secondary findings and the false positives. And here also the term non-actionable might also be a term to be on debate with, because what makes a disease actionable? Now you can, lowering the cutoff value, you can use another qPCR with different primers and probes. You can use a second-tier test. We tested epigenetic immune cell counting, so that's on dry blood spot that you do quantification of the presence of T cells, so not only the tracks but the T cells itself. And you can also play around with the referral scheme, so when do you refer a child or do a second test? So in this study we have 62 referrals and we had a cutoff value of 10. So what we did, we evaluated all the different options and see how the different options would reduce the number of referrals. Now, if you lower the cutoff value, then of course you have less referrals. And in every situation, we always pick up the SCID, so that's the good news. We also tested another PCR, and what we identified, the cases that were false positive, a couple of them had a SNP in the primer probe region. It's a commercial test, so we are not allowed to know the sequence where the primers and probes are located. But when we sequenced the region, the company confirmed, yes, this variant is in the primer probe region. So that makes sense to also have a backup qPCR. Also with the epigenetic cell counting, you also prevent referral of the false positives, and also the new algorithm. So then we have a second card. If your track is between the 2 and the 10, you repeat the track, and then for most of the patients, the T-cell numbers and the tracks we store in the first 2 weeks of life. So that's also an option. But still, I think we're not completely yet there. So at this moment, most of the programs work with track analysis, then referral of the child to the pediatrician, to the pediatric immunologist, then flow cytometry is done, and then genetics. Another model, I call it a Norway model. That's what they do in Norway, and unfortunately Asbjørn could not be here, otherwise she could present her strategy herself. So what they do in Norway, they do the track screening, and before referral, they do panel sequencing of SCID genes. And only after that, the child is referred. And then also you can do the flow cytometry and the confirmatory testing. So in this way, you can also be a more less strict with your cutoff value, but if you have the mutation, you are more sure about your referral. And you can potentially prevent unnecessary referrals. And also from our studies, we know that the impact of a referral on the family is huge, because you've given birth to a child, you think it's healthy, and if the screening is abnormal, you need to go to the hospital. So that's really, has huge impact. So I think we all need to prevent unnecessary referrals. And there's another option, and that's also what I would like to present here and hear your opinion about it. If you do a panel before referral, you could also consider doing broader panel for SCID and other EAE, and also include inborn errors for which no track-like test is available, for example, HLH. And after doing the genetic test, then the referral and maybe confirmatory testing. But after hearing all the stories about validation and data interpretation, what do you think about it? Do we need to want it, or is it better to leave that leave that aside? Kimberly?
I was going to say for better or worse, and I think I have some real concerns about it. The UK apparently is going to do a pilot of doing whole genome sequencing for 40,000 newborns to do this very thing, not just for SCID, but for metabolic diseases and everything else. I think there's some real ethical issues about what you do if you find something that you didn't intend to find, but also what you do that you say you haven't found anything, and you haven't found anything because we didn't look in the right place. We didn't know enough about the gene. There's another gene. So I have some real concerns, but it'll be interesting what that data over the next couple of years presents, because that's what the UK government, without understanding the ethics and the science, not that they understand much, is pushing.
Yeah. Other ideas or reflections or opinions?
Yeah, I have one. You could consider only reporting already known pathogenic mutations. So then you know you don't, so your cutoff would be less. Yeah. And then you know you miss for sure mutations which might be implicated, yeah. But then you're more sure about the ones you report.
Yeah. Yeah. Yeah.
Yeah. In terms of time of diagnosis, the first strategy versus the second strategy, how long it takes, the difference of the time.
For the second update, they can do it really rapidly. So it's within the 1 or 2 weeks. So it takes a bit longer, but not too much that it's a problem in the end for the child being referred. Sophie?
Thanks. I realise that you're not responsible for the Norwegian model, but maybe you know. I would be really concerned if a child with low TREX and a negative genetic screen was not followed up. So yeah. Presumably it's just to inform the evaluation of the baby with low TREX that they perform the genetic screen.
Yeah. Now in their paper they have really a good backup plan for really the low TREX values that these get referred. So I think they have a really good safe network, because that's also necessary. Yeah. Okay, I go to the next slide. That's a paper that was published last year, Appraisal of the Wilson and Junge criteria in the context of genomic-based newborn screening for
inborn errors of immunity. I think this is a really important paper because you say, okay, it's difficult, but the technology is ongoing, and you can say, okay, we're not going to do it because we see a lot of hurdles, which there are, but if you're not doing it and let it go, then the developments can also go in direction you don't want, so you don't are not in control anymore. And what they nicely did in this paper, they looked at all the classification schemes, and they also indicated, is there a treatment available? Because that's also important. If you consider genetics in newborn screening, you still need to keep in mind that there's what you find. Is there a benefit for the child? Do you decrease morbidity or an early mortality by screening for a disease? So this is also something to keep in mind. So I think what needs to be done is that, first of all, we need to develop the genetic approaches, so the technical approaches, also the analysis. But there are also the clinical aspects, also the Wilson and Jung criteria, and what is your clinical path? If you have the referral, how do you do in the lab the functional validation? Because in any case, you end up with a variant, and you need to prove that this disease causing, especially if it's not published yet. And the cost involved is also really an issue. And I don't know how it's in other countries organized. The screening is paid by the public health, and the diagnostic is paid by by different budgets. So if you put genetics in the screening, how does it work in the end for the costs? And the costs are really increasing if you implement screening, especially if you do it for all newborns. And of course, there are a lot of ethical, societal, and legal implications that needs to be considered. And the way what you do is also implementing on the social implication. If you screen only a certain set of genes, is different than genome-wide screening, that you have, in principle, all data available. How is it stored? How is it managed? And then communication, I think that will be key also to the public. What is the screening about and how does it work? So I think there's a big challenge for us, and I think it's important that we as a community also work on this and take the challenge to see what's possible, and also say, no, we don't do this, or we do only up to this point. So I also have a case. This is a girl. She was born May 2021. She had tracts of 8. And she was clinically well, and there was no maternal immune suppressive therapy, and the family history was also negative. And flow cytometry showed that she has T B-cell lymphopenia, but she didn't have a SCID phenotype. So at that time, it was decided to watchful waiting. And in August, September, the T and B cells were decreased, and she had high IgM. So genetics for a primary immunodeficiency panel was performed, and also antimicrobial prophylaxis was given. And here, as you can see, with the IgM, it was really high, and also the T cells were. Here you see they started with 1307, and they really drop. The genetic results came back, and there were 2 pathogenic ATM mutations. So it's a compound heterozygous situation. One was from the mother, and other inherited from the father. And then is the question, is this case a classical AT patient or a classical AT with a hyper IgM phenotype? So we did also some functional assays, and we proved that there was no protein present, and there was also no kinase activity. We also did some additional analysis for the B-cell subsets, and we found reduced number of naive mature B-cells, and also reduced percentage of memory B-cells. The Phi-beta repertoire analysis show no significantly aberrant or restricted repertoire, and the vaccination response was moderate. Now, coming back to the point about the hyper IgM, there's really a significant difference in this chance for survival if it's a classical AT patient or a classical AT patient with a hyper IgM phenotype. So then it was considered maybe if you would like to do a transplantation for a patient with AT, which is in itself also a matter of debate, then this child will be in a good situation because she's still young and there was a good donor available, a 10 out of 10. So that was an option to do. What would you do? What would you decide? Yeah.
So that's a difficult question, Miriam, and I think I'm going to get a lot of comments if I answer. But honestly, there is no established guidelines for performing stem cell transplantation in ataxia. On the other hand, I think our conditioning protocols are really improving, toxicity reduced. So I'm not so sure. It's a devastating disease. Of course, we're not going to cure the neurological problems by going into stem cell transplants. There's maybe in the future some treatment for the neurological devastating effects of the condition. But honestly, I find it very difficult, probably not go for transplants unless there's some very inspiring data coming up. But I would not go for transplant at this point. But not saying I would never.
I think you nicely reflect the discussions we had. And also when you look at literature, there are some studies, but N is 8, N is 3, N is 1, N is 1. So the overall survival, the first study was 25%. The other study was overall survival of 100% and this uneventful follow-up of 2 years. So it's not really a strong data on this. And we also had some communication with several people from the field. And you have also the ethical concerns about how the neurological disease you don't treat. And the child is very young, so that could be a pro, but also a con. So we decided not to do it and to wait. And then the hyper IgM restored. So in the end, it is, I think, what we know for now is that classical AT, and we're not sure anymore that's a hyper IgM phenotype. So she's now in the AT follow-up clinic, so she will be followed up carefully. But we decided not to do the transplantation, and in retrospect, I think it was a good decision not to do it directly. So it is also indeed what I concluded. So the conclusion is the identification of disease-causing variants in asymptomatic patients should be interpreted with care. So even if you do the functional validation and you have some immunological follow-up, you, especially when it comes to stem cell transplantation, is really a serious decision because there's no point of return. So this is also illustrating the difficulties of newborn screening. And my last sentence is that I think more research is needed to determine if and how to integrate genetics in newborn screening for now and also in future. I would like to thank colleagues from the LUMC. Dagmar, she was highly involved with the patient with AT, Robert Bredius and Arjan Lancaster, and from the laboratory, Maartje Blom. She just defended her PhD on the newborn screening in the Netherlands. Thomas, Ingrid and Melanie, and also Alexander Taylor from the UK, who did the ATM protein analysis, and Pim Vergeer for the public screening. I thank you for your attention.
Yeah, Miriam, thank you very much for this nice look into the newborn screening questions and challenges. Are there any further questions? Then, there's one.
Just a comment that in the discussions about the introduction of newborn screening by track analysis in the UK, there was a lot of anxiety about making diagnoses of AT because of the neurological aspects of the condition and the potential to undermine initial kind of bonding with the baby. And I just wondered how it was handled, sharing the diagnosis with the family here, assuming that did happen.
Yeah. She was, in this situation, we didn't have the AT diagnosed in the first week. Maybe that's, we found it only after when it was screened, so then the baby was 6 months old. So that would make sense, but when I heard Dagmar talking about it, she was so in shock that when she heard it was AT, she said, oh no, it could be everything but not AT. But the parents are really well understanding and also really open in the discussion for HSCT, and they understand the considerations, and they appreciate to have. I think in this situation, I'm not a clinician, but what I can understand is that the way you communicate with them and give them the explanation and information they need, and also let them talk with the expert from the AT clinic, I think that was really important. Because especially for AT, it's really, not all AT patients are picked up with screening, but some can be picked up, yes.
Okay, well, thank you, Miriam.
Oh.
And we're all longing for a cup of coffee. Yes. And see you back at 11:10. Now we have the young people also talking about challenging cases. So we hope again, like the last session, with lots of discussions and questions. And the one who will start with his case is Jorge Diogo Silva from Porto University Hospital Center in Portugal. And you go ahead.
Yeah, thank you. So good morning, everyone. Contrarily to I think most people here, I'm a medical genetics resident, and so today I'll bring you a case that's maybe a little bit more focused on the clinical aspect, but I think that your expertise will be very valuable in discussing some of the findings. And so the case that I bring is entitled "The Tale of Two Swords Between Immunodeficiency and Autoimmunity." So this is the family tree of our patient. So we have a 13-year-old girl. Both parents have a history of allergic diseases. She has a paternal cousin with Cornelia de Lange syndrome, and also the paternal grandmother has Besset's disease. In terms of pregnancy, this was uneventful. The neonatal period was also uneventful, and there was a normal birth anthropometry, but the patient developed fungal onphalitis during the newborn period. When we go into infancy, we see that while the patient had normal weight, stature, and developmental milestones were also normal, she presented a lot of signs and symptoms, such as recurrent oral, perineal, cutaneous, and nail candidiasis. You can see here the extent of these lesions at 4 and 5 years old, and all of these were responsive, fairly well responsive to antifungal medications. But she also had other symptoms, such as recurrent after stomatitis, which was very debilitating, chronic ulceration of the cornea, and also esophagitis and atrophic gastritis, which presented simultaneously and were both responsive to proton pump inhibitors. When we move on to school age, she had normal puberty and normal development of normal pubertal development, but she also developed some episodes of genital ulceration and also mild splenomegaly. I like to say that there was no history of any chronic or unusual viral or bacterial infections, so we were only seeing more fungal etiologies. Just some general analytical tests. We saw that several skin biopsies were performed, so they confirmed the fungal etiology. There was a mild elevation of liver enzymes, which was attributed to the chronic use of antifungal medication, and also a positive pathogen test. So the first question, which I think is fairly straightforward, is that what we think about what is the likely immunological defect in terms of immunophenotyping. And I'll ask some others that are more interesting maybe than this one. So in terms of immunological profiling, the myeloid lineage was perfectly normal, but we did see lymphopenia, more mild or moderate at least. And then when we look into the different cell lines, we saw normal B cells, NK cells, and CD8 cells, but we saw a decrease in CD4 cells and an increase in gamma delta T cells. We did also Th17 and T regulatory cell count, and they were also normal, and antibodies were also normal. But then we also did an autoimmunity profile, and we saw positive anti-nuclear antibodies, positive anti-smooth muscle antibodies, and we observed that the patient had a HLA-B51 haplotype. And so we now do the questions that are more interesting. What is the immunodeficiency pattern? And I think this is more or less straightforward for all of you. But this one may be a little bit more interesting. What is the autoimmunity pattern? Because we are seeing a lot of symptoms in here in the patient. Mm.
So I think maybe I would test autoantibodies against IL-17 cytokines to assess the cause of the fungal infections.
Mm-hmm.
And I would do genetic tests to assess whether it's a STAT1-GOF or a CARD9 deficiency.
Mm-hmm.
But it looks like STAT1-GOF patient.
Okay, yeah, I think that's a good answer. And oh.
I'd also consider TNF-AIP3 because of the sort of Bechet's aspects of the phenotype together with the autoimmunity, although the severity of the CMC would be very unusual.
Mm-hmm. Okay, that's also a very good point. Okay. Oh.
I would also cover a mutation in AIR,
Mm-hmm.
AIRE.
Okay.
And you can screen for that by looking for autoantibodies to interferon alpha,
Mm-hmm.
which is often positive.
Okay, thank you. Okay, so now just moving on. So what we did, so there are some very good answers. So we were thinking about that this was maybe regarding the immunodeficiency first, chronic mucocutaneous candidiasis, because it's a more straightforward phenotype we are seeing. We tend to simplify things. And so we asked for a gene panel with screening for CNVs for this group of immunodeficiencies. And in fact, we did see a STAT1 variant in heterozygosity that was being described as a STAT1 gain of function. So the diagnosis was established, and I think this is very straightforward and not really the point of this case. The point is the other diagnosis, which we are thinking on Behçet's disease. So Behçet's disease is an autoimmune vasculitis, and what is very difficult for the clinicians is that there is no serological marker for nothing regarding Behçet. So we tend to focus on clinical criteria. And the patient actually had the general clinical criteria, which are, you can see here on the left, because she had recurrent oral ulceration and also two additional factors, recurrent genital ulceration and a positive pathogen test. But in fact, she also had eye and skin lesions, but the eye lesions were not the most common for Behçet's because they are usually uveitis or retinal vasculitis. And the skin lesions it was very hard to assess because of the continuous fungal infections. If we look at the pediatric Behçet's disease criteria that has been proposed, we need 3 points out of 6 at least for establishing a diagnosis, and we only had undoubtedly recurrent oral apoptosis and genital ulceration, with the skin and ocular involvement being a little bit more hard to assess. So we established the diagnosis of the immunodeficiency and likely Behçet's disease, but I think we are fairly confident that this is the diagnosis. And so what we're thinking regarding the STAT1 gain of function, can this explain both things? I mean, the immunodeficiency is very well established, but can it also explain Behçet's? Is it an independent finding? And how can we think in terms of pathophysiology? Maybe I'll leave the discussion open in the end. because I'll add more information now. Just to get your thinking going. So in fact, when we look at the Th1 and Th17 responses, when one is overactivated, the other tends to be inhibited. And in terms of the naive CD4 cell differentiation, if we have a STAT1 gain of function, it is possible that we have cell-mediated autoimmunity, which might be the case here, and also the loss of function of the decrease in function in Th17, which cells which are important for extracellular fungi defenses. But there are some oddities. We did not see any changes in serum IL-17 nor in the Th17 cell count, which was interesting. And that although we know that this gain of function has been associated with several autoimmunity manifestations, it has never been definitively associated with Behçet's disease. And most of the autoimmunity are more in the endocrinological disorders. In terms of future perspectives in treatment, we are now considering the JAK inhibitors, which are still ongoing in terms of being tested regarding efficiency, but the patient has a lot of functional impairments regarding the food infections. They are very debilitating. Otherwise, the general condition is good. And there's also the possibility, obviously, of hematopoietic stem cell transplant. And so this is maybe this is the last slide, and I think this is the most important part of the discussion. If this STAT1 gain of function is solely responsible for Behçet's, it is an additional factor. Is it independent? Because we do have the HLA-B51, which is an haplotype of susceptibility to this condition, and we also have the family history of the grandmother, so there's also another point there. And as I told before, these autoimmune disorders that have been described in this gain of function are mostly hypothyroidism and type 1 diabetes mellitus. And there's not a lot regarding rheumatological conditions. And another point of discussion, I think, is if we start treatment, either JAK inhibitors or do the transplant, will it also improve the Behçet's phenotype? And this is just regarding, so the variant was affecting the coiled coil domain of STAT1. So in terms of long-term prognosis, they tend to be a little bit better. But once again, regarding autoimmunity, we don't know anything about this. So this is just the acknowledgement slide. This is part of our genetics team. And I think I will leave these questions as for the discussion, which I think this is like the most important thing to discuss here in the case.
Thank you.
Okay.
So who would like to open the discussion here?
I was just going to say we have a couple of patients with STAT1 gain of functions on JAK inhibitors.
Mm-hmm.
Some have done really well. Interestingly, we can use tyrosine phosphorylation of STAT1 sort of we've actually used it to sort of titer out the optimum dose for a couple. One's had to go up to quite high doses because they were failing treatment at lower doses. Okay. We also deliberately transplanted 3 girls with Behçet's disease, no underlying genetics, because the ulceration was so bad and their quality of life was so poor. Mm-hmm. We don't know what the underlying cause was, obviously. They've done really well, but I can't speak for all of them. Yep. I do worry in that we have one child who is a STAT1 gain of function. Her father is as well. His father unfortunately died in his mid-30s, so you do worry he didn't have and he was actually on JAK inhibitors at that time. Can you keep these kids on JAK inhibitors their whole lives because of the susceptibility to other infections they're going to get by doing that? I think it's definitely worth trying. It may completely change your quality of life, but whether it's a long rest of your life treatment, I'm not sure.
Yes, I think that the pediatricians that we work with, they tended to not start the JAK inhibitor, tend to delay it as much as they can because of that, because she had some points where the infections were more or less controlled, but now the food infections are really bad in terms of, and then in terms of functioning of the quality of life is starting to decrease, so that's why, but that will always be something dynamic in the long term in terms of when to stop, when to start. So, but thank you.
Thank you for the presentation. So I have a question. Did you test interferon signature in this patient? Because maybe can represent a kind of link between explaining the inflammatory vascular phenotype, and can also be useful for choosing the therapy to control the symptoms.
Yes, I don't think they tested. Not that something that I was not. I'm not aware that they did. So I think that's a good hypothesis. Now that we join all of the trying to join all the puzzle pieces. So thank you.
So thank you. So I'm pretty sure we have, at least in our cohort, 2 or 3 patients with STAT1 gain-of-function mutation and basal disease. Mm-hmm. So, however, I don't know about the HLA. Yep. And regarding the bone marrow transplant, so it's a question we have had for almost more than 10 years. Should we go for bone marrow transplant or not? We have plenty of patients who are doing pretty well. Yes. Plenty who are not doing well. The problem is when we go for the bone marrow transplant, it's usually when we go because we have a number of patients who are doing well, and we're thinking, okay, let's wait. And when we start the bone marrow transplant, they are a very bad disease. So I guess it's why it was not working. But we still don't know. The JAK inhibitors, so some are reporting fantastic effect, the others are reporting it's getting worse. So I think it's still a difficult situation, and sometimes we're thinking, okay, maybe we should go for bone marrow transplant before they do a severe disease or not. But I will check and send you an email regarding our other cases.
Okay, thank you. I was actually, just to answer the one part of the comment, is that I saw some reports of STAT1 gain of function where they listed the autoimmune profile. And some had they were suspect for Behçet's disease because they have oral Eftes disease, but they were not definitive, so they just checked one of those 6 criteria. So there was somewhat misleading in the reports. That's why I was like, is there any, or is there any, or isn't any report on this? So I think that the link is fairly it's understandable. So I think we're going somewhere with this. But thank you for the comment.
So if there are no further questions, we thank you very much.
Thank you.
And we continue with Lucia Pacillo from Bambino Gesù Children's Hospital in Rome.
Okay, good morning everyone. I'm Lucia Pacillo I'm a pediatrician from Bambino Gesù Children's Hospital and a PhD student in immunology at Rome Tor Vergata University. So I will present this clinical case of this young girl who's now 22 years old with a negative familial history and an eventful pregnancy and birth. And she was first evaluated at the age of 1 for severe atopic dermatitis and several episodes of anaphylactic shock. She presented at 3 years old an HPV negative papillary hypertrophic hyperplasia of larynx and tongue that resolved. And from the infections point of view, she experienced recurrent respiratory infections, in particular bronchitis and 2 episodes of pneumonia that didn't require intensive care and that get proved over time. Her clinical history is mainly characterized by a severe atopic phenotype, as already mentioned, a severe atopic dermatitis early onset, onset, and severe food allergies, multiple severe food allergies, for which she is on a very restricted diet, free of many kind of food, milk, egg, fishes, beef, all vegetables, that of course affects her quality of life. Moreover, she has a severe and refractory asthma, allergic to many inhalants, for which she's on chronic therapy with antihistamine and inhalator, steroid, and LABA. From the infections point of view, she has got chickenpox when she was a child without clinical problems. Then she got infected by ABV without clinical problems, but it remains a chronic low copies ABV replication in the blood with complete seroconversion. More recently, she also experienced gastrointestinal symptoms, in particular alternation of diarrhea and constipation, for which she underwent a full endoscopy that showed the lymphocyte and monocyte infiltrate. And moreover, we also detect a thrombophilia state with upper homocysteinemia and homozygous MTHFR mutation. So what investigation would you perform? Maybe I'll answer this since it's a very easy one. So we of course did a full blood count that show normal leukocytes and lymphocytes count, but show also hypereosinophilia. Serric, IgG, IgA, and DGM levels were normal, whereas IgE were very high, especially at the beginning. Maybe with a diet, the restricted diet, their level went down. Also, RAST and ISAC tests were positive for all the antigens she was allergic to. She has no specific response to vaccines, but actually she didn't complete her vaccine schedule since she was for the anaphylactic shock history, and she has a good response to chickenpox that she got infected with, and also she has a complete seroconversion to ABV, as I already said. In terms of immunophenotype, she has a normal frequency of T cells and also distribution. Also regulatory T cells were normal in frequency, and also circulating TFH cells, as you can see here, they are normal in frequency and also in PD-1 and ICOS expression, as well as normal NK cell frequency. But we detect a reduced frequency of Th17 cells in the patient compared to healthy donors. And in terms of B-cell phenotype, she always had reduced B-cell frequencies since she was a child. This one is referring to one of the last phenotypes, so is referring to young adults. And as you can see, she has reduced B-cell frequency and reduced memory B-cells, in particular with switched B memory cells. Moreover, in vitro B-cell differentiation was low. So at this point, with this clinical and immunological phenotype, what would you suspect? Any ideas? It's just atopy, a very atopic girl, or could it be something else? Okay, I'll go ahead. Maybe it could be a primary immune deficiency associated with the high IgA levels, so also with allergies. So these are hyper-IgE level syndromes by one of the last US classification, and also many of the characteristics, even if very less profound, make us think about, okay, deficiency. So at this point, with this immunological clinical phenotype, how would you proceed? What do you do next? No one. Okay.
I hear mumbling, but when someone wants to use the microphone.
Okay. Okay.
So maybe do the panel for the hyper-IgE syndrome genes, or go directly for the whole genome sequencing, whole exome sequencing.
Okay. Of course, genetics was the first things we did. But that's the point of this meeting. But in the meantime, we also did functional tests. So I'll give you the answer later. So this one is the docate expression by FACS and by Western blot on ABV B cell line and on the B cells from PBNC that resulted reduced in the patient compared to healthy donors, and also in all B cell subpopulation. It resulted reduced also on K cells, but not on T cells. Both in CD4 and CD8, docate expression was normal. So the NGS gene panel for primary immune deficiency resulted negative. So we proceed to clinical exome sequencing that disclosed an heterozygous de novo mutation in STAT6 gene, not reported in NEOMAD, and predicted, actually at the beginning was predicted likely benign, just 7-8 months ago, and then the verdict now is pathogenic. And we also verified that it was a germline mutation. So, just a brief summary of STAT6, that is the main transcription factor that mediates biological effects of IL-4 and also IL-13 that are necessary for the type 2 differentiation of T cells as well as B cell proliferation and class switching to IgE production. So an hyperactive STAT6 signaling results, of course, in increased Th2 differentiation, elevated IgE levels, and all this process, of course, cause the allergic inflammation, asthma, and as well as atopic dermatitis and so on. So we started to look at the literature, and we couldn't find anything about this mutation except for a link between this mutation as a somatic mutation in up to 13% of follicular lymphoma, and also STAT6 polymorphism association with food allergies predisposition. But until 1 month ago, there wasn't anything else, and only a month ago it was published this preprint from a Canadian group about two patients who presented a clinical phenotype very similar to our patient with a severe atopic immune dysregulation, and with an heterozygous STAT6 mutation. In particular, patient 2 is the mutation in the same amino acid, even if it's different, and they also did functional studies that established that both variants cause gain of function phenotype associated with enhanced phosphorylation of STAT6 and stimulated also after IL-4 stimulation. So, at the moment, we were doing also a proteomic analysis with other patients, and we have seen that this patient had high levels of IL-4 that fit with this phenotype. Ongoing autoimmune done are CD23 expression, that is a STAT6 target gene, and also STAT6 phosphorylation, and we are in contact with this group to do other studies. So it's a work in progress. The open question I want to discuss today is we have seen that docate expression was reduced on B and K cells in this patient. What could be the link? Could it exist a negative feedback loop or something like that? And moreover, we have seen that somatic mutations of STAT6 is connected to follicular lymphoma, which could be the link between STAT6 mutation and malignancy. Could our patient be at risk of malignancy in her life? And of course, at the moment, what's the most suitable target therapy for the patient, also to get better quality of life? In the past, it was hypothesized omalizumab since high IgG levels, but knowing the pathway, the alternative pathway, we can also suppose dupilumab, for example, that acts on IL-4 receptor, or maybe also. And in this preprint, one patient is on dupilumab and is going well. And our future perspectives are to demonstrate STAT6 mutation pathogenesis, as with the functional studies I've mentioned, and of course to expand the cohort of genome function STAT6 patients in order to clinical and immunological characterize this new novel primary atopic disorder. And I want to thank all my supervisors, colleagues, and you for the attention. Okay, thank you.
Thank you very much, Lucia. Could you go back the slide with the questions? Does anybody has any answers or suggestions?
Any thoughts?
I have the question concerning the Western blot with DOCATE. Did you test only one control? Because you say that it's reduced, but it could be in the variability of... we don't know if controls can have a huge variability in DOCATE expression. So if you compare only to one control, you see a reduced expression in your patient, but it's possible that if you test several controls, you will see the same pattern than the patient?
There were several controls, I think. Okay. There are 3 controls here. We compared it with several controls, actually. Okay. So.
Any more suggestions
any
or questions?
Thoughts about the link of DOCATE and STAT6? Because there is the link with STAT3. I don't know if it could be similar. Okay, I got step 1. And what do you suggest about therapy? Okay, no.
Yeah, I'm sorry, I can't answer any of those questions, but I just wanted to mention that we have one base pair downstream, we have exactly a similar mutation. So, and we're also in contact with the authors, and it was a patient originally coming from Hong Kong. So it really would emphasize further that it's a gain of function and something very domain or amino acid residue specific.
Yes, he told us that there are also other groups who are contacting him.
So maybe more frequent.
Okay.
I have a question in the chat, but because the chat is occupied apparently, via from Evelien Zonneveld, and she asked if you consider DOC8 MLPA, deletion CMV testing.
Maybe we can. It could be also a deletion DOCATE. I don't know, because the DOCATE is a difficult gene. We didn't do.
So that might be something to consider then.
Okay, thank you.
I want to ask
just a minute.
It's true that we have control where it's not decreased DOCATE expression, but it's my question to you or some people that work more on DOCATE, if she has a low B memory cell, and I don't know if the phenotype could, it's in some PID with similar phenotype, DOCATE could be reduced for other things. This is, I don't know if there is a link. We still, is an open question. So but in your in your hands, there is a high variability in DOCATE expression in control. It's just a question. I think, okay. No, usually we do Western blot or fax analysis of DOCATE to make a diagnosis faster, because for the problem that frequently there is a deletion that you can escape. So it's
okay. Thank you. So in the chat, there is I'm sorry. So, Xiaopeng is saying that human gene connectome servers suggest a connection via RAC1 and MAP kinase 8.
Okay. Thank you. Thank you.
Oh, there's still one more question. Could you?
Just a quick one. I didn't manage to see if the other patients reported had any viral problem, any viral infection similar to HPV or any other virus.
No, they have more gastrointestinal issues. One of them had PEG. And also vascular problems. One patient died for cerebral aneurysm. And so that's another point where we have to look for in in our patient, because as I mentioned, she has thrombophilia.
And what about lymphoproliferation?
Lymphoproliferation, actually, no, she has not large spleen or lymphadenopathy. She has just this chronic IBV, very, very low copies, like 120 copies, but persistent with the complete seroconversion. But she doesn't have symptoms of this infection.
Okay, thank you.
Thank you.
Okay, if there are no further questions, thank you very much, Lucia. And the next speaker will be Georgiana Maddalena Orsu, as well from Bambino Gesù Children's Hospital in Rome.
Okay. Good morning to everyone. I'm Georgiana Maddalena Urso, and I'm a medical biotechnologist at the Bambino Gesù Children's Hospital, working in the primary immunodeficiency unit. And today I will talk about this case of a female patient that was born 15 years ago from non-consanguineous parents and presented with a negative family history for primary immunodeficiency. This patient was admitted to our center with severe recurrent infections, such as pneumonia episodes that required hospitalization, skin infections caused by molluscum contagiosum, and so important was a really severe lymphopenia, although with a normal sad blood count. And a normal, the immunoglobulins were in a normal range. This patient manifests with a combined immunodeficiency, and serological analysis reveal mainly this positive count of antinuclear autoantibodies. But although all of these characteristics, the patient have a good vaccine response. Going deeper in the immunological analysis of this patient, a really increased amount of naive T cells was found. With, on the other hand, a reduced fraction of central memory and effector memory populations compared to age-matched healthy donors. And so, interestingly, a low level of recent Thymica migrans population was revealed in the presence with a higher increase of the CD31 negative, CD45 array positive cells, and this is so typical because this population normally is under a percentage of 10, and also regarding to the RTE, I have to say that the track quantification was normal. and these exclude probably a thymic problem. Also, this same T-cell phenotype identified on the CD4 positive cells was also revealed on patient CD8 positive cells. And going ahead, the B-cell phenotype also in the patient exhibited a reduced amount of memory B cells, also unswitched and switched memory B cells, while the other related population to B cell phenotype were in a normal range. What else could we explore at this moment to better check this patient? What do you think?
I would check for the CD45RO to check it's not the RA polymorphism.
Yes.
So could you repeat the answer then of the question? So the audience online can hear it.
To check for the CD45 arrow because is the next step in the differentiation of CD45 from the CD45 array marker. This is? No, quite.
But when you do when you check for naive central memory,
Yes.
effector memory, and use CCR7 and CD45RA,
Yes.
there is a quite common polymorphism of CD45 that cause the retention of the A exon. So it looks like all the cells are RA positive.
Okay.
So it looks like that when it happens. So I would check RA versus RO to see if the pattern is like that, or you adapt. Sometimes the memory T cells are actually RA positive.
Yes, we do. After this, also the check for the differentiation of the population. Okay. Okay. Any other? Oh.
I think you can. You can continue.
No? Okay, I can go ahead. After this, we check also for the TCR expression, and it was normal, as well as the quantification of the Treg cells in the patient was in a normal range. The only piece of the puzzle that was so wrong was the T-cell proliferation in response to different stimulus. And at this point, we can ask help from molecular investigation. Which genes could be involved, according to you, in these clinical immunological features? No one? Okay, I will show you. We firstly checked for some genes by Sanger sequencing that could be related to this phenotype, but all were negative. And for this reason, we go ahead with whole exome sequencing that revealed a new variant in an interesting gene that is TREP150, also named as TREP3. And this variant was reported as uncertain significance, has a controversial pathogenicity scores among the different prediction tools, but it is really, really rare. And for this reason, we decided to understand if there could be a link, a correlation between this mutation and the clinical immunological findings And checking in the literature, we found that TRAP 150 has an indirect role in the signal-induced alternative splicing of several genes. But interestingly, for our case, it has also an indirect role in the splicing of the CD45 gene, encoding gene. What happened? That in resting cells, TRAP 150 is able to bind a phosphorylated form of PSF, which is a factor, a protein splicing factor. And in this way, this protein block the splicing, the mRNA splicing. While when T cells are stimulated normally, the unphosphorylated form of PSF isn't able to link with the TRAP 150. And is free to do its activity of its splicing activity on the CD45 gene, and in this way is able to induce the splicing of this gene and the differentiation of T cells from naive to memory cells, and thus the activation. At this point, how will you proceed for this in this case with TRAP 150 mutation segregation, or maybe with the protein expression or other functional assays?
Anyone? All three of them, maybe?
Yes. Of course, we have to proceed with all of three. And first of all, we search for the segregation, and we found that also the father has the same mutation, and in fact, also the father had referred to our clinicians that he also had some episodes of recurrent infection in his pediatric age, but over time his condition was became pretty good. Moreover, looking at the protein expression, the father exhibit a lower protein expression than compared to the healthy control, although a little more higher than his son. And at this point, we have to ask what about the father's phenotype, because he also has the same variant. And when we checked for the T cell phenotype of the father, we found again the same increased amount of naive T cells and the reduced amount of memory cells, thus confirming again this block of cells differentiation. But to go more in deep, we also go to see and to analyze the mRNA of CD45 protein. And we found that both in the father and in the patient, and both in the stimulated PBMC with CD3 and CD28 beads, as well as stimulated with the PHA for the generation of T cell lines. In both these cases, when the cells were stimulated, they not only exhibit the Ra isoform, which is the first isoform of CD45 gene encoding for the naive T cells, but also this additional isoform, that is the array form, including also the exome 5. The exome 5 is one of the first exons in these genes that is spliced out, and this is curious. For this reason, we saw also in by flow cytometry how this population differentiate through the both marker of differentiation, CD45 array and AR0. And we found, interestingly, that not only the patient that I show you here, but also the father, had this intermediate, this middle population of cells exhibiting both markers compared to these healthy donors that normally differentiate to the memory phenotype. And this was found not only in the PBMC stimulated with beads, but also in the derived T-cell lines. Okay, at this point, finally, what we can see, that this patient has a very severe immunophenotype with a persistent lymphopenia, increased naive T cells, low memory T and B cells, and a typical high population with CD45 array positive and CD31 negative, as well as an atypical middle population of CD45 naive and memory B cells. This patient was included in the European profound combined immunodeficiency. And in view of the mutation identified in this patient in TRAP 150, we hypothesize that probably this mutation induced an arrest in this link between TRAP 150 and PSF that avoid the splicing of the CD45 gene and thus a block of cell differentiation. But this phenotype could also improve over time, because in fact not only the father became so good during his life, but also the patient now exhibit a good clinical phenotype, although he persists to have this severe immunophenotype. And what we could do, maybe for more understand and define this particular case, probably a co-immunoprecipitation of these two proteins, or a CRISPR-Cas9 knock-in knockout of the protein, or what else? And I want to thank all of you for your attention, particularly my supervisors, Caterina Cancrini, Giulia Di Matteo, all of my colleagues, and again, you for this opportunity.
Well, thank you very much, Georgiana. Are there any remarks or questions regarding this case?
So it's difficult. So I would try to restore the phenotype by transducing the cells with the RITA allele, because so it could be convincing or not. It could be the good one or not. And my second question, so have you performed whole exome or whole genome sequencing on on the trio?
No, because this was a whole exome sequencing that was made many years ago, maybe around 2050, and the NGS was really, really young technology. And at the time, no.
because maybe if you consider that the father and the patient are affected, maybe it would be trying to see if there is any variance,
additional variance, maybe
shared by both of them.
Yeah,
that couldn't. But I would try to complement a phenotype just to be sure that.
Okay. Thank you.
Regarding the infectious phenotype.
Okay.
And if I understood correctly, the patient presented just warts, right?
Yes.
The warts were vulgar warts, flat warts, and they were chronic in the time, or they revert. And also if the father has similar infectious phenotype.
I don't know to say this really well. Maybe.
Chronic, but not so severe, and now he didn't have. The father, I don't think. So is an old case that we have we didn't work in the last year because from a clinical point of view, they go well. There is an improvement. So we have not a lot of energy to understand more. It was very intriguing for the phenotype, for the mechanism, and for the phenotype, because usually CD4, CD31 positive cell correlate with mostly with the TREC. In this case, the immunological phenotype is clear in the father, but we are. So it's not so severe, the clinical phenotype is severe in the infancy, because they refer to us from another hospital to ask if or not, if do or not bone marrow transplantation. So we did all the check-up at the time, immunological, and we see that was a severe lymphopenia, but not alteration in B cell, in a humoral compartment. He has a good specific antibody response. We put at the time in prophylaxis with Bactrim, but we didn't start immunoglobulin, for instance. So the clinical was not so severe.
So if there are no further questions, and we can thank you very much.
Okay. Thank you.
And we continue with Francesca Conti from Bologna. That's great.
So good morning to everybody. Thank you for this opportunity and to be included in Young Fellow, but it's not so true. And I will talk on behalf of Cristina Cifaldi in the lab of Caterina Cancrini, that we made all the experimental plan. I just sent and suspected the patient as clinician. Voila. So today I will talk to you about an EBV positive Hodgkin lymphoma in a pediatric patient. So let's see what's behind. Let's start with the pedigree. The patient was born from non-consanguineous family. He has now pretty 8 years old, from Kosovo, and he has 4 brothers and 2 sisters, and both parents and brothers and sisters are apparently healthy. Nothing to say about pregnancy, delivery, oxological parameters, and neonatal course. The age of onset of its medical history was at 5 years old with lymphoproliferative disorder that will deepen now. And just in its past history, we have a dental abscess in 2017 without other history of recurrent or severe infectious disease. So he had a diagnosis after a pretty year of lymphoproliferation without a really criteria for the diagnosing an Hodgkin lymphoma at the end of 2019 with the detection of EBV in the tissue, in the nodes. And for that he was treated following the Euronet PHLC2 protocol, and here you are the drugs used. Only after 2 years from the treatment, colleagues in oncohematologists send us the patient because the detection of, because they realized that he has an hypogammaglobulinemia, but was tested only after 1 year after the chemotherapy. So we don't have the test before this huge therapy, and he undergo IVIG replacement therapy. He has also persistent EBV viremia, ranging from about 10,000 to 100,000. In 2021, he starts again with a lymphoproliferative disorder that at the end of that was biopsied multiple times, and just at the end of 2021, he has a real criteria to say that was a relapse of the ABV positive Hodgkin lymphoma that was currently treated with brentuximab because the relapsing, with poor response after currently 5 doses. It's not a complete cycle, but however the PET is not a complete or a partial response. So let's see about something about its immunological workup. Here you have the extensive immunophenotype and immunoglobulin dosage. We highlight that he has a high percentage of B-naive cells, pretty absent or absent B-switched memory B cells, low IgG for the age, in particular low IG1, and this immunological workup was performed 1 year after the end of chemotherapy. So low IgG and absent switched B memory. So we have a patient with Hodgkin lymphoma, a relapsing of this diagnosis, with also lymphoproliferation, EBV triggered, and with persistent EBV viremia, and lack also of complete ABV seroconversion, hypogammaglobulinemia, especially IgG, and absent switched B memory cells. So first question, because we don't have the T0 before chemotherapy, do you think that this immunological alteration may be secondary to treatment, or you consider, however, as potential primary hypogammaglobulinemia, primary antibody defects? Just yes or no.
You can't know because you don't have the pre.
Yeah, it's difficult to say. So because the phenotype, however, is highly you have a high suspicion for primary immunodeficiency. So this is the second question. You have to consider that there is a part of a possible part of iatrogenic hypogamma, but also that you don't stop here. So if you think that there is an underlying inborn error of immunity, which category, which kind of you will suspect? Yeah, easy. So in front of this suspicion, so EBV susceptibility, inborn errors of immune system, which genetic approach, how I will approach from the genetic point of view. So you can Sanger sequencing some selected candidate genes, they are not so much, NGS target panel, CGH array or exclude CNV copy number variation, clinical exome sequencing, whole exome sequencing, or whole genome sequencing. How?
I would first go for NGS targeted panel.
Okay, so, right. But in our case, we proceed because we have I don't have the possibility to proceed rapidly to NGS target panel, and we have primers for 3 X-link, for 3 genes associated with EBV susceptibility and EA. We decided to rapidly sequence MACT1, SH2D1A, and XIAP that resulted wild type. So we proceed to NGS target panel, and we send to Rome to perform the analysis. This is the gene analyzed, and this is the results. So an heterozygous deletion on PIK3R1 gene that is known to be associated with APDS2, that also is an immunodeficiency associated with susceptibility to EBV. So you can think that, wow, you have the diagnosis, but it's not so easy because this deletion is confirmed on genomic DNA, and it straddles the P50 isoform of the gene. It's reported. There is equivalent deletions reported in literature. In Varsom, is predicted as likely benign. Is not so rare, and is not in a conserved region. So our question is this a significant variant, even if can fit with the phenotype? And so which test you perform at this step? You will consider, however, because it can fit with the phenotype, you will go further? No? You stop here?
So I would not go further with this one. Anyone else? Anyone a different opinion?
I probably would investigate it.
Yeah.
I do something functional to investigate it because it just fits the phenotype
However,
so well.
yeah.
Even though in silico says it's benign, I don't think you could ignore
I agree. So even if there's, it's not really at the threshold. Yeah.
And do you have any information on the frequency?
In the
there. Okay.
Is there? Yeah. So for the European, it's it's a. So, however, we proceed because we want to be sure that this variant couldn't have any impact on the phenotype. Also, because lymphoproliferation, EBV, B-cell perturbation could fit with the APDS2 phenotype. So, in the immunology lab in Rome, Cristina and Giola performed expression experiment, and effectively they saw that both on EBV cell line and T-blast, P85 isoform were overexpressed compared to healthy donor in 3 different experiments, while P50 were reduced, not absent, but reduced both on the 2 cell type. And so we go ahead, and because we are investigating this pathway, what would you expect from the functional data if the variant had an impact, an increase of phospho-AKT, an increased PS6? No. We test it. It's difficult to say a priori. And the phospho-AKT evaluation and basal PS6 were comparable to healthy donor, but we know that some APDS2 it does not exclude completely the diagnosis, the hypothesis. What we know about the possible genotype-phenotype correlation, what we know about P50 isoform, we don't know so much. We know that it's a isoform involved in insulin and glucose metabolism that can be regulated by STAT3 in a mouse model and a specific mammalian cell line. It was reported in 2012 a case of a patient affected by high gamma globulinemia, absent B lineage, completely lacking P85 subunit, with unaffected other isoform, but they show at least the expression on the different cell line of P50, P55, so add some information about this isoform, not on function. And in 2019, a paper showing in human breast cell cancer the CAPG protein as direct activator and factor that promote the transcription of P50 regulatory subunit. So in conclusion, we have a patient with hypogammaglobulinemia, absent B memory switched cells, EBV susceptibility, and lymphoproliferation. We found for the moment only this heterozygous deletion straddling the 5' UTR region of PIK3R1 affecting P50 isoform. Oh, sorry. And at the experiment performed, for the moment, we see some impact that we have to confirm on expression, not on hyperactivation of the pathway. So for the future plan, first of all, segregation analysis that we don't have yet performed because can answer to many questions, cDNA analysis, TA cloning, real-time PCR. If the segregation analysis doesn't show any the same heterozygous deletion in parents, we go further with protein expression on different cell lines, time course of AKT phosphorylation, maybe cell death proliferation analysis mediated by STAT3, and also CAPG mRNA level to see if there is some correlation with the overexpression of P85. And just for curiosity, glucose and insulin metabolism in the patient, and eventually bone marrow biopsy to see the B cell maturation stages. So thank you for the attention. It's difficult to trust on it, but it's difficult to live completely.
So thank you very much, Francesca. We already have some questions behind you.
Sorry, I've got a question and a comment. So I guess my question is, you have something that may or may not be real, are you go ahead and do, because you only did a panel, is it worth still doing whole genome sequencing on this family to see if there's anything else?
Yeah, I think.
The comment was going to be that, I think a lot of us have seen the very common alanine 91, valine, and perforin by itself is fine. It doesn't do anything by itself, but you put it with a second pathogenic mutation, and I think a number of us have seen late onset HLH because of that, and that's present in sort of 2.3% of the population. So, is this a predisposing factor, and is there a second hit somewhere we're missing in the same pathway or something else?
For sure.
But I was asked about
for sure. Mostly after this amazing workshop, but it's just we try to proceed with the available tested. However, we have also the clinical question that the patient developed a second hit of the lymphoma. It doesn't respond well to the brentuximab, so we will try with sirolimus and let's see what happens. It's difficult to.
Did you take him to bone marrow transplant because he clearly?
Yeah, the first, but it's difficult without a diagnosis at not so severe phenotype. Even if to hits of lymphoma, it's not.
We recently were referred a very similar child. It turned out that his recurrence was genetically completely distinct from the first tumor, which was proved by fingerprinting. So it was a second primary and not a recurrence. And that helped to build the evidence that it was a primary immunodeficiency. And we got the diagnosis there by staining for SAP protein, which was absent, and he had XLP.
Okay, nice to. We will check also. Yeah, thank you.
I mean, I find it really interesting because it's one of these cases that sort of it's almost sort of you almost want to exactly to continue like this. But genetics or population genetics almost argues against to spend more time on it because it's unlikely to be I guess what I find intriguing is your reduction of the P50.
Yeah.
Because that is a hallmark of P85 deficiency, because it stabilizes sort of the whole complex. And if you have P50 sort of as a secondary deficiency, and just to see this, I mean, it's probably a lot of work. It could fail.
It's
but I think to introduce your variant in a cell, in a cellular system, where you can replicate this phenotype, that sort of is probably sort of the next step. Relatively easy, just as plasmid, and then to see whether you can affect the P50 level.
Yeah, for sure. Yeah, I think I agree.
Just by curiosity regarding the familial segregation, you were able to complete the familial segregation
of all brother and sister. We will complete. We do not perform for the moment. We plan to perform now in the next future, and we have the possibility to test every member of the family. So let's see, because they are a lot of brother and sister do not apparently have any phenotype though. So
and it will be great if maybe you can also some of the experiments that you performed to also to see.
So some family segregation is the first step, sure. So, thank you.
So the next presentation is an online presentation of Evelien Zonneveld-Huisson. So we are going to set up the system for this. So a few minutes.
So go ahead. Thank you.
Thank you. Well, thank you for this opportunity to present my case.
Wait,
One second.
Wait. We try. We have a last try.
Okay.
She should get. Oh my God. Sorry.
No better option right now. Sorry.
That's okay. Just tell me when I can start.
Wait. So I'll try to. No. Okay, I put the max.
Yes.
Okay, let's go. Sorry about this.
Oh, no problem. No problem. I'm really glad that I had the opportunity to show you this very clinical case, which is really unknown yet. So I'm actually hoping for you to help me further on this. I don't have that much interactive questions in between, but I really hope for your interaction at the end of my presentation in maybe helping me further with this case, which has a pretty unusual combination of features. And it considers this 20-year-old male. He started off in his life with a very mild, actually, hypogammaglobulinemia, which started at the age of 5 years, with low IgGs and low IgAs, with even elevated B cells and normal TB subsets, for which he receives IVIGs because he has recurrent upper airway infections and ear infections. And the IVIG really solved that problem. And they have never stopped actually giving him that. Then he has this story of a colitis, which actually, when you really ask through, starts early in childhood already with severe abdominal pain and some blood loss in his early childhood years, but then, well, that disappeared. Well, the abdominal pain stayed, and eventually now, recently at the age of 18 years, really an ulcerative colitis has emerged, and it's at the moment very difficult to treat. And then next to that, he really has a symptom that I cannot link yet to the rest of his symptoms, but he really has a hypercalciuria from the age of 14 years old. And secondary to that, there is a severe nephrocalcinosis, and secondary to that, there's a partial diabetes insipidus and anurosis because of the huge amount of volume of urine he produces. There's a slight developmental delay. He had some motor and speech delay in the early years, but now he has a normal intelligence. But he's somewhat lower level than his parents. There's some behavioral problems which are quite severe. The PDD narcissistic autism spectrum disorder that he suffers a lot from, and he also has signs of ADHD. And there is this ectodermal dysplasia, which is characterized in this patient by hypodontia, hyperhidrosis, and a very dry skin. And then when you look at his family, you see some symptoms recurring in his family members. And I try to give them more or less the same color so that you can recognize which is what. His mother is quite similar to him. She also had ear infections, but we recently tested her Ig levels, and her Ig levels are really normal, so that she doesn't have the hypogammaglobulinemia. She does have a nephrocalcinosis, and she also has an oligodontia, which is even severe, more severe type of the hypodontia that her son has. His other brother also has hypodontia, and maybe the autism spectrum disorder is inherited from the father, as he also suffers from this behavior problem. The boy has a quite distinctive phenotype, and I got permission from the patients to show you his pictures. I cannot connect all the bits and pieces together, but I just show them so maybe you see some connection between what you see from the boy and his immunological phenotype. When you look at these pictures, you can see his long neck on the left-hand picture. On the middle picture, you see his eyebrows are quite prominent. There's a little bit of upslant of his eyelashes. He has a broad and short nose, a quite short philtrum, and when you look at his pectus, you see it's a little bit, we call it pectus excavatum. I'm not sure what's the English word for it. And there's some striae on his skin. I don't show the oligodontia here, but he's really missing a lot of teeth in his mouth because they didn't erupt in the adult phase. And then a colleague of mine has seen this boy before in the years 2015, 2016, and did already a lot of genetic testing. A SNP array was performed then, which showed a maternal deletion, which does not contain any omen coding genes, and was considered then and now for re-evaluation considered still probably innocent from the new variant. He had this duplication of chromosome 16, which nowadays is recognized as a risk factor for developmental delay, behavioral problems, and also aortic dilation because it contains the MYH11 gene, and we advised cardiac surveillance for that. And you see a lot of gene panels being done in the former years, all based on whole exome data, using different panels to analyze. And we included MLPAs in that time because the CNV analysis on exome data wasn't performed yet. And eventually, after the 3 gene panels or more 5 gene panel sequence, then the whole exome sequence was analyzed in trio, and then no disease-causing variant was found. So they said, well, you have to come back. So when he came back with me, I made my own differential diagnosis with the things we know now. And I could think actually of 3 groups. It could be, of course, a primary immune deficiency next to the other problems that he has, a non-syndromal form, for example, defect in plasmablasts or a CVID kind of clinical phenotype. He could have a syndromal primary immune deficiency because, of course, all the ectodermal features we thought of NEMO, IKBKG, or something else in the NF-kappa B1 pathway. And then you can search and see if other features could be combined. For example, the hypocalciuria, nephrocalcinosis, you could find combined with hypodontia in the 2 syndromes I mentioned at the bottom. And actually, I couldn't find any connection between nephrocalcinosis and PIDs. But if you have, I really like to hear about that. And then, I repeated a lot of genetic testing, but I also tried to do it a little bit different than before because we were also, of course, interested in maternal variants for some features. The PID panel I did, including maternal variants. I had to ask for a separate sequence analysis of IKBKG and MLPA because it's really incompletely covered in our PID panel. The PIK3C2A sequence analysis had to be done separately because it's not a PID disease. and all the gene panels for developmental delay and early onset IBD I did in a trio de novo analysis, but the gene panel for ectodermal dysplasia I did including the maternal variants. And there's still one gene panel analysis ongoing in nephrocalcinosis, and I will only do this on the data of the patient itself. So as far now, I don't have a diagnosis. We could, of course, repeat the whole exome sequencing and trace back into the variants there. But I really would like to hear from you in which direction we should look. Or do you have any suggestions to add, for example, to high differential?
So, thank you very much, Evelien. They are doing some technical stuff, so that's why it was a bit silent, but we could hear you properly. And,
so I wonder whether you have thought about checking the NF-kappa B pathway function.
Could you hear the question?
For me, it's quite impossible to hear that. Can somebody repeat it?
Sorry, I haven't listened to the question. Sorry.
I wonder whether she thought about testing for the NF-kappa B pathway function.
So you were talking about testing the NF-kappa B function.
Well, we haven't done that, but if you also think in the same direction, that could be something worth considering. How would you do that?
Testing for the NF-kappa B the NF-kappa B pathway?
Mm-hmm.
Yes, so you can look at a whole range of pathways like TNF response. There are a number of pathways that can be tested.
So you could assessing the NF-kappa B pathway. So looking at the TNF response. So I guess by P65 phosphorylation, I-kappa B alpha degradation, translocation of NF-kappa B into the nucleus, so things like this, I guess? Yes.
Thank you. That's a good suggestion. I'll take it.
And maybe also there is, so as you said, there is a problem for NEMO sequencing in the NGS panel because of the pseudogene in the region. So maybe trying also to sequence NEMO in cDNA from the patient, mother, and brother.
Did you hear that or?
I think you suggested to sequence the NEMO gene in the cDNA of the patient or in RNA.
Yes.
Yeah, thank you. Yeah, that's something worth considering as well. So you also think a lot of NEMO here.
Hi, Evelien. Very nice effort. I was wondering whether you checked in more detail the TP63 gene. Isn't that also with this dysplasia phenotypes, or is it ectodermal dysplasia and also tooth agenesis and some clefting occasionally? But I think the clefting may not always occur. So I think there is some cases with isolated oligodontia as well. So the gene is P63. P63 or TP63. The little brother of P53. That is also sometimes described, if I'm not mistaken, with oligodontia and some related features that you also shown. So maybe there's something in that gene.
TP63. I wrote it down. Thank you. I'll dive into that.
And maybe consider somatic mosaicism of NEMO. You can maybe isolate T cells and see if it carries some variant there.
Have you heard about this?
No, can you repeat?
So somatic variation, it's for NEMO because it might be some somatic mutation. So.
Yeah, but then it's strange that the mother has the ectodermal features as well, don't you think? Then it would be a germline in the sun, and the mother with some mosaic, but then a mosaic in the sun is pretty improbable, I think. If it would be a mosaic system in the sun.
Are you sure that the deletion, the maternal deletion and the paternal duplication together could be done something? No.
that I didn't hear.
The deletion, the maternal deletion, chromosomal deletion, and insertion, the paternal insertion, because the patient received from the mother a deletion and from a father the insertion, the duplication. So, what do you think about the possibility that this chromosomal alteration could do something in this patient?
Okay. Yeah, well, I really looked into that, but I couldn't find any connection with the genes that are involved in this duplication and deletion. And the duplication is actually a quite common one, so it's not really suspected of a translocation, or, for example, in one of the parents. So I couldn't find any connection with the found CNVs yet. Do you have other ideas about that?
Maybe I missed something, but did you test also the X inactivation of the mother, and did you look what was the X that was inherited by the two brothers? Because it could be also, I guess, a clue. You can see whether it's possibly X-linked for sure because of the two brothers affected. So maybe looking at the X inactivation of the mother and the X that was inherited in the two brothers.
Mm-hmm. Yeah, no, we haven't tested the brother yet. That's really something worthwhile considering, because the ectodermal dysplasia may be the main feature in this family. Maybe that's the direction to go now, to map that phenotype with the genotype. Yeah. And then we can do a kind of a trio analysis with the patient, the brother, and the mother, maybe. Looking at the X chromosome. That could be a really good strategy to follow. Yeah, I agree.
So few suggestions from the chat.
Yeah.
So is there any hypophosphatemia?
I have to look that up. I don't know. Why do you ask?
And EDAID could also suggest ORI1 or STIM1. The former being a little bit less well covered on exome.
Mm-hmm.
And did the patient have any evidence of proteinuria, renal insufficiency that could have led to secondary hypogam?
Oh yeah.
So this is. So, and just finished. This is always Xiaopeng and saying this and asking, because atypical Fabry disease can be a cause of non-specific GI issues and secondary CVID finding that is often missed in adults.
Sorry, that last, what is it still concerning the proteinuria, or is it another comment? Because I couldn't.
So asking, Xiao Peng is asking because atypical Fabry disease can be a cause of non-specific GI issues and secondary CVID findings that is often missed in adults.
Oh, Fabry. Now I see. I don't know. I have to look into this.
Okay, Evelyn, I think you have a lot of suggestions and you can work further with this. Maybe you get further with this case. We hope for you. There are no further questions? Then thank you very much, Evelyn, for your presentation.
If you can have a look in the chat, there is many suggestions on the chat.
Oh, even. A new suggestion. Oh yeah, I'll see. I can read the chat now, so I will take it from there.
Perfect. Thank you.
Thank you so much. I'll stop sharing now.
So, yeah, I have the name.
So we cannot do anything for the sound, correct? Good, good, good.
Can you hear us? No. Ha, la, 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 la. We cannot hear you.
You're on mute.
Okay. No, we cannot hear you.
So, in the
Is it
meantime,
working?
yeah.
Yes. Yes.
Okay.
Good, good. I'm very glad. Okay. Thank you very much. Am I good to start?
Okay, so wait 2 seconds. So here I'm introducing Shugniver Niklen from PacBio, who is going to talk a little bit, present a little bit on PacBio and I think HiFi sequencing. So I guess go ahead. Can you share your presentation?
Yes, of course. I just need to know that. Let me just share my screen. So bear with me a second. Oh, it's not allowing me to share. Why is it not allowing me to share? Let me try again.
Okay. While she's starting up the presentation, I would like to thank all the young fellows from the AGP for their case presentation. I think they deserve a warm applause from us. And thank you very much for this nice interactive session. And we wait a bit for the PacBio presentation, and afterwards we will have lunch.
That's the green
and
button.
To continue talking, the afternoon will be dedicated to the patient perspective with some very nice lectures and interactive sessions.
Okay. Wait. Yes, go ahead and try to speak loud, please.
Okay,
thank you.
Thank you very much. Hello, everyone. My name is Savinda Niklen. I am relatively new in PacBio, and I joined them just over a month ago. There are so many exciting things happening here at PacBio, so I'm pleased to be speaking to you all. I wish I was physically in Paris, but I've learned firsthand how COVID hasn't quite finished and things can change at the last minute. So this is a short presentation to give you a very high-level overview, and I'm very happy to discuss more individually, or my colleagues, of course. So over the last year and a half, PacBio has changed considerably. We still have the major product being HiFi sequencing that you see here, which is known and loved by many, and we've added a growing portfolio of new sets of products. So nanobind DNA extraction through the acquisition of circulomics. So why circulomics? It's really easy to explain. If you want to have the best quality sequencing, you need the best product to be put into the workflow, and they have great products to use. Also, you see here, sequencing by binding SBB. This was through the acquisition of Omniome, a short read sequencing company. Now, it may be a strange thing to do for a long read company to be acquiring short read, but there is an explanation for this. From a biological perspective, DNA or the RNA may already be degraded. So there are large amounts of work being done in this area. For example, in my area of expertise, looking at a mother's blood to see the fetal genome, or for example, in oncology, looking at taking biopsies to see the early signs of cancer or indeed the progression of cancer. And in these types of applications, the DNA is often 100 base pairs or smaller, so short read works really well. What you're going to see from us is a growing, expanding group of products to help address the requirements that you have within your work. And we have over 50 products currently which span the entire workflow, including sample preparation, instrumentation, consumables and software. The other point here is we are growing the number of people within the company right across the world, including in Paris. Hopefully you have met Benjamin and Anne Lynn who are with you. And in addition to more staff, we also have our European head office in London, which opened a month ago. And it's a great space to visit. A short trip on the Eurostar, and it will take you there, and a short walk from the St. Pancras station. So we'd be very welcome to accept you there. So
For those in the audience that don't know us, I want to talk a little bit about what hi-fi sequencing is. If you look at the graph on the left, it shows you what is great about the technology. It's accuracy plus its read length. The real differentiator here is the ability to take a 25 kilobase fragment, high molecular weight DNA, to then circularize it, as is shown here, and the DNA polymerase will make continual passes. Each time there is a single pass, the information gathered is collated, and as we go through it multiple times, we remove errors, and in comparison with the short read sequencing, we have a higher degree of accuracy at individual read level when compared with Illumina. You can see here on the latest NovaSeq platform, and of course with Oxford Nanopore. This is compared with the Q20 chemistry, and it's almost an order of magnitude lower than PacBio. It is this unique combination of long reads and accuracy that gives us the ability to give the most accurate and comprehensive genomes of any sequencing platform out there on the market currently. I want to show some 2020 data here comparing the sequencing platforms. We're very proud that PacBio hi-fi sequencing has the least errors in sequencing of the whole genome. This is because we have high accuracy across all the major variation classes, so it's structural variants, indel calling, and single nucleotide variants. In comparison, if you look at the others, Illumina do well at indel and SNVs, but often are challenged with the large structural variations. Oxford Nanopore do well with the structural variations, but single nucleotide accuracy is challenging, and of course indels too. To have comprehensive accuracy with a single technology, PacBio hi-fi sequencing is really the only choice. One additional point I want to talk about is repeat expansions. I'm sure it's known, more than 40 diseases, mostly those which affect the nervous system, are caused by expansions of simple sequence repeats dispersed throughout the genome. These repeat expansions are very difficult and almost impossible to sequence with short read sequencing. PacBio hi-fi sequencing can fully and accurately resolve these types of variation, even at very low coverage, so 5-fold, 10-fold whole genome sequence coverage. I'm going to talk about the 3 pillars shown here of human genetics, and we all know integrations of omics is key, so I'm going to start with the genomics. So here you can see some of our partners in the States. Hi-fi sequencing is of course used in a research setting, but we are now seeing this move more and more to clinical settings. In North America, we're working with the large children's hospitals that you see here. Hi-fi sequencing and hi-fi reading has had a huge impact on providing answers for families and those with critically ill children. Many of them will have gone through years of diagnostic odyssey. And the key take home here is if you're using PacBio hi-fi sequencing and comparing with the outcomes from short read sequencing, it allows you to identify significantly more structural variants of all classes. But what also surprises people is that with this long read, we're also able to call the structural nucleotide variants. So, to recap, we have individual read accuracy, which allows us to call these, but the length allows us to read regions of genomes which are not usually sequenced by the short read sequencing. In the Children's Mercy data here that you can see, roughly 200,000 more SNVs were called with the PacBio data, and they were also able to do the indels, phasing the genes into haplotypes, so determining if the alleles were maternal or paternal. It's not just across the Atlantic that we're working in. We've got an increasing number of European collaborators. Earlier this year, we announced the one with Genomic England, looking at those families who've had short read sequencing already and yet still don't have an explanation of the causes. And sadly, almost 75% of individuals who went through the Genome Project in the rare disease space still don't have an answer to what's causing their disease. Now, we've just finished sequencing in the pilot project with them, and we're looking at the data and we're hoping to announce the output very, very soon. We are aware that not everybody is doing whole genome sequencing, and we're targeting and collaborating with partners for targeted sequencing. In China here, you can see on the left-hand side, we're very excited to be working with Berry Genomics, and they're taking the thalassemia assay through the Chinese FDA at the moment. And also, we have a collaboration with Twist Bioscience. The first of these collaborations is around pharmacogenomics, and you can see here a pharmacogenomics panel. If you are going to the ESHG in a week or so, you'll hear a little bit more about this. And people are increasingly using PacBio sequencing across more and more areas of biology. So you'll see this as the area develops. Now going on to the epigenome, the second pillar of genomics. It's critical to understand this in terms of what's happening for human biology and health. And I'm going to give some examples of some new areas that we are working in to show you how this works. So here you can see within the last few weeks, we launched a new version of our software, which has allowed us to deliver some of the promises for sequencing that we made a few years ago. As the polymerase goes around the template, we can look at how long it takes to incorporate a base and some of the kinetic characteristics of that incorporation. And through some machine learning tools, we're able to recall the incorporation of a base adjacent to a 5-methylcytosine. So wonderfully, you can get a 5-base genome with no additional cost and complexity. And you can see here it includes the 4 bases, 5-methyl, the long read phasing, and gives you very, very high mappability. Now I'm going to talk about the third pillar of human genomics, that is the architecture and complexity of the transcriptome, i.e. gene expression and transcript isoforms. Interestingly, in terms of the transcriptome, people and researchers have been predicting since 2018 that it's going to be transformational in this space, as you can see from this quote here, showing that there's going to be a shift, a paradigm shift from gene-centric to isoform-centric within this area. A major contribution to this is the PacBio isoseq method. Now, looking at the isoseq method on this slide, you can see many of you will have used short read sequencing in the past and are looking to characterize the different isoforms and the genes that are expressed in the genome. Normally, it would mean lots of shotgun sequencing, typically 150 base pair fragments. And if you're lucky, some of them might splice between one exon and another. And if you're really lucky, you might get those exons on one read. Then what you do is statistically define what it looks like. So in reality, it's probably not very accurate. We know that there's a lot of important biology in the isoforms, and it really does determine, for example, how one cell differs from the other. With the isoseq reads, you can see here at the bottom, what we get is the full length. We can read from one end of the transcriptome to the other end and understand exactly which isoforms are expressed. This is going to allow us to understand the biology at the RNA level far better. If we then layer that with the single cell work, which you can see here, and this is a growing area for us in this world, historically what you would do is capture the ends of the different transcripts and try to determine what cell types they come from and what is present. But you're missing the rest of the biology. You're missing the isoforms that are expressed within the different cell types, and this information is available through the single cell full length RNA-seq. Now, let me just go through to the next bit. There's a lot of work going on in this area, and there are developments happening, very exciting developments. An example here is shown as a preprint from the Broad Institute, and it's a new method called the mass isoseq, and it dramatically improves the yield of the single cell isoseq reads. Until now, the single cell isoseq projects would need multiple smart cells. The Broad have developed a clever method where throughput is increased by concatenating the fragments. Now, the reason for this is the smart cells can take very, very long fragments. So by concatenating the smaller fragments that are involved in this area, you can actually increase the throughput hugely. So there's a 15-fold throughput increase. There's very robust cell type clustering, which you can see from here, giving you a very clear picture of what's happening. And also there's a 30-fold greater discovery of the differential spice genes. So I think this is all without the need for short-read RNA-seq, so a really key area. And the expectation is we will see more and more development for this as this technology is in the hands of yourselves and your colleagues. Now, in summary, I hope I've been able to highlight some of the applications that are relevant. You can see here a long list of the areas that our technologies are available in and used in. Of all of these have been made on the PacBio Hi-Fi sequencing on the Sequel 2E system, and many of the examples I've shown today are invisible or inaccessible with short-read sequencing. I'd like to thank you for your attention, and I'd like to thank the organisers for allowing me to speak. But if you do wish to know more about this, please do reach out to Benjamin and Anne Lynn, who are there with you in Paris. And of course, if you do want to visit our European head office to talk about projects that we can perhaps do together, or want to see our technologies firsthand, do reach out to ourselves. Thank you very much for your time.
Thank you. So, just in case, because we have trouble with the connection and with the sound, so except if someone has questions. So Benjamin is here, so he's at the end of the amphitheater. I'm sure he will be happy to reply to your question. Is there any burning question? No. Thank you, and we'll try to see you soon in Paris. Hopefully, you will recover soon. Thank you.
Thank you very much. Thank you. Bye-bye.
Bye.
Very important subject, patient's perspective. So thanks all for staying, and I want to welcome Julie Powell as the patient representative, and Martine Pergien, that's a difficult name to pronounce, to start the first session, and after that Marco Crimi will present more about platforms. So take the floor.
Okay. Hello everybody. This is the awkward session of the day, just after lunch. Everybody just feels like they want to have a wee snooze and everything. So I'm hoping that I keep your attention long enough, like that you'll learn a little bit. So I have been asked to talk about the patient experience, and sometimes I think, maybe using the word patient can be a little bit off. And I prefer to use the word lived experience wherever we can. So, where am I? Here, this one. No. Which, this one. There, I've got it. I'm not very technical savvy here, I'm afraid. Okay. No, I don't need it.
You don't need it?
No, I don't need it. No, thank you. So, who am I? So my name is Julie Power. I'm a housewife and a mother. I'm a retired occupational therapist, and I'm living with a rare disease since 2005. 2005, at the age of 40, I was diagnosed with Wegener's granulomatosis vasculitis. It took me a long time to learn to say that. And then they changed the name to granulomatosis with polyangiitis vasculitis. So it took me another while to learn that. But I was previously a very healthy woman, and I was in my dream job. I was in my forever home, and life just suddenly changed, literally changed overnight. A few years down the road, took me a while to meet somebody else with the same condition as myself, but a few years down the road, after taking my head out of the sand, I started looking for support. And there was no support groups, so I decided, right, we'll set up one. And we ended up that we have set up a support group for the whole island of Ireland for any of the vasculitis conditions in Ireland. And I'm the present patient contact and policy officer of that group. In my journey, I had very aggressive vasculitis. And at the beginning, I was lots of different treatments. Like I was told, there's no cure, but all the treatments are quite toxic and they have quite a lot of side effects. But I seemed to kind of go through these treatments, you know, like fairly quickly. And eventually I ended up on this really new drug, but they weren't sure it seemed to work. It was designed for something else. But we didn't know what the long-term effects of it were. But this drug worked for me. And I thought, God, that's great. And then I started thinking, well, what happens then whenever this drug stops working? What's going to happen? So I started getting a little bit interested in research and trying to find out what can I do as somebody living with the condition, what can I do to help the research process. So I got an opportunity to join UPATI and do a UPATI course, and that's a very intensive course, learning about all the ins and outs of therapeutic intervention, and about clinical trials, and about all the different ins and outs of research. So I became quite interested in that. And I am now the vasculitis patient lead in ERN RITA, and that's how I have come to be here. And I thank Anne and Marielle very much for the opportunity to talk here today. So that's me. And why am I here? I am here just to talk about the impact of molecular research and the lives of those living with rare disease, not necessarily vasculitis, but any of the rare diseases, or in fact, any condition, because we keep talking about rare disease, but I mean, it can be any condition. And I want to also talk about how the lived experience can add to the whole research process. So my agenda today is we're going to have introductions. So I'm going to turn to you now shortly. I was going to use Mentimeter, but last minute we weren't able to use it. So we're going to go live. And then we're going to talk then about patient involvement, engagement, centricity. These are words that are bandied around all the time, but just trying to kind of define what do they mean, what are they? And then why have a patient involved in the research process? And then how does molecular research impact our lives? And then talk a little bit about real experiences, rare experiences as opposed to, and how can we improve patient involvement? So this is where I'm going to turn to you, and I'd like to know who you are within the audience. So perhaps if you're a researcher, can you put your hand up and let me know? So we'll see how many researchers. Well, quite a lot of researchers. If you're a clinician, can you let me know? Is there any clinicians? Yes, clinicians. Any policymakers here? No. Any engineers? One. Any patients? Well, actually, we're all, in some way, we are all kind of patients. Well, any other? Is there anything that I have missed that hasn't captured what's in the room? No. Okay. Okay. Okay, and what can you let me know what you're kind of expecting this afternoon? You know, like from when you heard there was going to be a patient perspective, what were you kind of thinking this might be? Is anybody going to be brave enough to? Yeah. Yeah, yeah, that's good, good. Anybody else brave enough? No? Okay, okay. Well, hopefully we'll get to that. So these three words that I was saying to you, patient involvement, patient engagement, and then patient centricity. So I kind of think, just imagine there's a big circle, okay? So the big circle. So we're going to talk about patient involvement, and that's kind of just inside the perimeter. So that patient and public involvement entails research being carried out with or by members of the public rather than to or about them. So that's like you're involved. The patients are involved. So then patient engagement is with research goes further than participation in it. So you have engaged individuals and communities who initiate research, advise, challenge, or collaborate with researchers. Their involvement is always active and they have a crucial influence on the conduct of the research, on its design or methods, products, and dissemination or use. So this patient engagement would be, kind of midway between the centre and the perimeter. So that's patient engagement. And then patient centricity is from the middle out. So that's putting the patient first in an open and sustained engagement, and respectful, compassionate to achieve the best experience and outcome for that person and their family. So basically, the patient will be an integral and valuable partner in the research. They'll be respected as an equal stakeholder. And it means that the research can deliver meaningful outcomes, not just what's interesting to science. So, back to you. How do you think patients are involved in research? Yeah. I think, for a lot of us, the research where we present with the problem. We, we'll consent to having our data used or whatever, and then at the end of it all, hopefully we have a solution. I think that's kind of like the traditional way at the moment that patients are involved. There's more leaning now towards having patients involved throughout the whole process. And this, to me, is a much more healthy way of having patients involved. So there are 5 myths about having patients involved in a research process. So the first myth is that the engagement would be too expensive, be far too expensive to have, patients or carers involved. The second myth that citizens aren't up to it because, we don't speak the same language, the medical language or that we're not up to it or that we're not going to understand all the concepts. Engagement only works for easy issues. And this is one of the things, when you're having a community involved in the whole way through, yes, there will be issues, but by the time you have resolved those issues, you have a really good piece of work. That's been completely transparent. It is going to meet the end, the end needs. It's going to be a meaningful piece of research. Citizen power is a floodgate we should avoid at all costs. And I know, yes, for some patients, whenever they come into this kind of a process, if they're early on in their journey or they're having difficulties in coping with their journey, there can be a time where there's a lot of time spent, talking about their story. And that can be seen as, there's time not being wasted, but this is taking longer than it should do. But we can get through that. And citizens don't want to be involved. They just want good service. And, it's surprising how many people want to know much more about their condition. And I've heard before in the rare disease community that what is it? The FBI wouldn't have anything on a mother of a child with a rare condition, because the mother will investigate absolutely everything down to the nth degree. So there is we're very engaged in what is happening. So the benefits of having a lived experience involvement, the outcomes can be massively beneficial, including better study design, much better recruitment numbers and process, much better retention rates, better protocol adherence, and it's also empowering and educating us, the patients, to manage our condition better. Okay, so here I am going a little bit off-piste, and I didn't want you falling asleep, so I decided I was going to try a few different things here. So, so I'm using Prezi. Just hope it works. So basically, this is I want to take you, like the journey, how the patient can be involved in the whole research journey. This is going to take a while.
Do you need some help?
It's just taking time loading.
Oh, okay.
No. I think I'm just going to lift my own laptop here. You can pretend there's lots of really interesting images going on in the background. Did I close it? Here we are. Okay, so this presentation was called "Crossing the Chasm to Lived Experience Centricity," and it was lots of explorers going up a mountain. So, so this first step was setting the research priorities. So, you asked, what do we want as patients? But the thing is, we sometimes we don't know what we want as patients. Sometimes it comes out in conversation. For us, for our community, one of the things was, of course, you want a cure, or you want a definitive diagnosis, or you want, effective treatments. But one question that kept coming up in our community was, how do I know I'm in a flare? And that was something that, we all live with it, and we all get confused with it, because whenever you think you're going into a flare, you're never sure, is it an infection? Is it, maybe you've overdone it the day before or whatever, and you think, right, okay, it's an infection, you have an antibiotic, that antibiotic doesn't work, so then you have another antibiotic and that doesn't work, and at this stage you're a couple of weeks down the line into your flare, and you could easily have some damage done at this stage, because you're not having treatment. So there's valuable time wasted. So this was one thing that was identified by our community, and the researchers took it up. It's whatever. It's just going round and round.
This one.
No, that's this one here.
Why is that? Here we go.
You see, it just keeps. I think it has to download. It's okay. It's okay. So that's, so establishing unmet needs, so listening to the patient community. So with us then, the researchers took that on board and decided, right, how are we going to to figure this out. And as a result, they started looking into biomarkers and, and what, they have identified one protein in the urine whenever we're in a flare, CD163. And that has been life-changing for a lot of us, anybody that has kidney involvement, because you can get access to your treatment right away. With just a dipstick and you've access to your treatment. So that it's just listening to the community. A lot of the time we don't know exactly what we want. Then the next stage then is, whenever there's the research project has been set up and everything, it's the practicalities of the research. So, how many times does somebody have to come to the hospital? Do they have far to go to the hospital? If they have mobility issues, do they have far to go from the car park to the lab to get their samples done? Or, are they in employment? Are they in gainful employment? Do they need to take time off for this? Or do they have childminding? Do they need, to arrange that? So it's the practicalities that are involved that we need to take into consideration. Another thing would be like risk benefit. For a lot of us, I suppose the benefit, outweighs the risk. Sometimes we will, we're prepared to have something to have the benefit. I suppose, I have an example later on that I'll share with you. With that. So then at the design and planning of the research project, so big area is consent forms, having them, that they're understandable for the patients, in their language, that they're clear, that they're not too long, that, and then do you give a consent form whenever you mention about the research? Or do you say, right, this is we're thinking of this and enrolling you in this, and do you want to take time to go away and think about it? Do you give them the consent form to take away with them so that they can talk to their family or others, about it? Or, do you say, right, leave the consent form, it needs to be filled in here, but, think about it and come back to me, this kind of thing. Patient information leaflets, again, that it's all in a language that's easily understood. We need to have somebody with a lived experience involved in things like the ethics review. We're all very aware of things going terribly wrong as regards ethics, and we need to have people involved at that stage. And the protocol synopsis, the design of the research, is there going to be further visits needed? Is there going to be, what else is going to be expected of the patients coming in? Getting the information out to the participants in a particular type of research. And that somebody with a lived experience, it's not just the person that's living with the condition, but I often think the family live with the condition as well, and they know nearly as much as the person that's waking up every day with that condition. So that they're involved in places like the investigators meeting and data monitoring, making sure, the patients know exactly what's happening to their data, and that they know that they can opt out at any time, that there is the option to be forgotten. And to be involved in study reporting. So again, that this is all transparent for the patient community going forward. And then things like the post-study communication, how do you get this information out into the community again? And being involved with the regulatory affairs and the health technology assessment, that those processes, that there's patient engagement there. What I was going to talk to you earlier was there's a hypothetical situation where for psoriasis there was a drug developed, and this drug did exactly what it set out to do. It was passing all through stage 1, phase 2, phase 3, got through to the HTA, was approved, everything. And whenever it came out, when it was being prescribed for the patients, there wasn't another drug that did the same thing, but whenever it was being prescribed for the patients wouldn't take it up. They preferred the old drug that they had. And the reason was this new drug got rid of the itch. But what was important to the patients was that they had no rash on their face or their hands. And that was the important thing for them. So, you know, like there was all this money spent, you know, like on this piece of research for, you know, a drug that was going to take away the itch. But at the end of the day, the patient population weren't going to be using it. Not all of them would have not been using it, but they preferred that there would be no rash on their face and their hands. And just to be mindful, you know, like in any research, that there are meaningful endpoints, that this is going to have an impact on someone's life. And that the whole process has been transparent. Like Marielle would have talked yesterday about the human phenotype ontology. I got it right. And I think that is a brilliant idea, and I think, you know, like the patient community should be invested in that and learn the ontology, learn the same phraseology that is being used. If that's made accessible to the patient community, I think that'll break down barriers. We end up with engaged participants and then reduced waste of resources. So that's the how that I would suggest that we can use for patient involvement. Now I have to try and get back here now. I'll close that. I'll just close that one. No. Oh, thank you. Okay, so I'm not starting all over again. It's all right. Don't panic. Yes, I want to show you this short film now about patient involvement, patient centricity, and it's done by, I can't find the sorry. It's done by a girl called Jenny Preston, and I'm sure some of you will know Jenny. She works in Alderhay Hospital, but she's very involved in patient and public involvement with the younger generation, young people, and getting them involved in research projects. So
okay, we're going to close it. I'm sorry. Do you see the mice in the map?
Yeah, it's on the other screen. So press escape.
Wait,
escape. Ah, yes.
Oh, yeah.
No, that's okay. There. This one here. I want.
You don't want.
No, I just want this one here. That's it. Yes, yes. So here we are. So this is just a little film. So you don't have to listen to me for a wee bit.
It's not. Wait. No, it's not that. Julie.
Huh?
It's not that.
Oh, it's not come up.
It's not even working. Escape now.
Aha. It's this one I want. Is that it now? Brilliant. Thank you. Thank you. Okay. Now I need to get it to start. I've lost the wee thing down at the bottom there.
It's coming up.
There's
difference with patient involvement by
starting to get the patient involved right from the beginning. So when you know that you're going to be doing a study, then bring the patients in right from the beginning. When will we ask for consent for this study? When will we start asking people for information? What kind of information? Get the patients involved right from the start with every step of the way.
What patient involvement should look like should be that patients are involved from the very, very beginning. And one of the things that we've discussed as a family is that really patients should be being asked, what do you think research should be?
Yes, because they know a lot about the condition because they've had it all their life. So I think they can put their feelings and input about it and see that we could research this part of the condition and then this part. So because I've had a heart condition all my life, I think it would be easiest for me to input some information than just like a researcher who's just finding out all this stuff.
We're living it every day, so to us we're maybe more sensitive to certain images, we may not want to hear certain words, we might find some things slightly patronising, and when you're living it day to day, you want to be taken seriously.
I also think that patient involvement in research, good practice, it has to be real. It mustn't just be tokenistic, where you're just asking patients at the end of a clinical trial to look over something, so it's just a tick boxing exercise. I think it's really got to be real, it's got to be done together, and it's got to be a really good collaboration between the patient and the clinicians and the doctors.
If we can change some of the things of how it's addressed to make it more friendly, then I feel that you would get more people taking part, and if more parents take part in studies, then obviously you're going to get better outcomes and the more trials that can go ahead.
For several years we've been working with pharmaceutical companies to try and encourage early involvement in protocol design. Fairly recently we had a pharmaceutical company come to us and they wanted to involve and engage with young people and parents on a particular study. So they came to our group and they looked at the protocol of a potential study. Our young people contributed to that discussion and looked at the outputs, the outcomes of the study and the design, so the practicalities of that study and the information requirements needed for that study to be a success. It was a fantastic opportunity and we've been trying to encourage that for several years. So it's not just at the patient information stage of the design of the research, it's actually at the very beginning where the most influence can be made. It was a great success. I think the pharmaceutical company were really pleased with the feedback and have used that to inform the next stage of the protocol design. And we hope to share that learning and share that good practice with other pharmaceutical companies as a standard of good practice.
Okay, now reduce here. I think I'm actually going to show another. I hope this one works. Another short film about a young fella called Owen, and just this is the impact of the kind of work that you do, the impact that it can have on somebody's life. How did I get it? There. Okay.
Is it white?
Yes. Half. Both white and black, I did.
White and black? Yes. He was probably around 6 months old when we first had a few health concerns.
Dirty.
At the same time that was happening, we started to have a few concerns about his development.
Happy birthday to you.
He wasn't sitting up, and then at about a year he wasn't walking and he wasn't even beginning to babble or talk. So that's when I had real concerns.
Come on then. So she's going. Come on, Owen.
It's hard for him in terms of his movement and his coordination in particular.
Big step again, Owen, look.
And he's quite insecure about movement, so often Rob and I have to be with him to do most activities.
You okay?
He can't talk very well. He's got limited verbal capacity, so he can say maybe 20 or 30 words.
Right, come on then, let's get on this way. Higher or stop?
He was on growth hormone injections for a while. He's had blood transfusions in the past. He's had a steroid trial, a lot of appointments. Some of them are quite invasive. We were told sometimes people just never get a diagnosis, which is obviously not an easy thing to hear. And it was at that point that we were entered into the 100,000 Genomes project.
We didn't know what to expect. We didn't know if we would get a diagnosis, and we'd been entered into studies before and we hadn't got anything.
We all have about 5 million places in our genomes where our DNA vary from each other, and this is what makes us individual. Owen had low blood counts, and as part of his investigations into that, he was seen at a specialist clinic at Imperial College Hospitals. Owen's results came back, and there was only one variant that had been prioritised by the pipeline, and this was in a gene called THRA. Initially, we weren't sure that this was really relevant for Owen, but we did know that Professor Chatterjee in Cambridge had a specialist interest in these very rare thyroid conditions.
We discovered resistance to thyroid hormone alpha in 2012, a condition due to a genetic mutation in one of the thyroid hormone receptors, thyroid hormone receptor alpha, and this is a protein in the cell which controls the actions of thyroid hormone on different tissues in the body. This condition is very difficult to diagnose because although patients don't respond properly to thyroid hormone in their tissues, blood levels of thyroid hormone are virtually normal.
We received the diagnosis when Owen was 4.5 years old. For me, it was transformational. I didn't realise how much I wanted a diagnosis. You put your life on hold and you think about, well, what's going on with my child? And you have no answers. And that's really hard to deal with. And then when we did get some of the answers we wanted, it was amazing because I knew it wasn't something I'd done.
Owen was initially referred to us when this condition was identified. And when they initially came, they didn't know very much about the condition because it was so rare. There's only about 30 patients identified worldwide. So we were able to share our expertise to inform them more about how the condition was affecting Owen.
To get that specialist support and information so we can understand what might happen for Owen in the future, it was a gift.
We started him on a thyroid hormone tablet called thyroxine. We reassessed him then in the clinical research facility to try and determine how he had responded to the treatment. And rather gratifyingly for us, we were able to determine that many features had improved. So he was growing, his energy expenditure, also termed his metabolic rate, was improving.
Help, please.
The treatment that he has at the moment is very, very easy. It's taking 1 tablet a day, essentially, which he takes without any trouble. There we go, good boy.
Owen really likes to go to Cambridge to see the team there because they have such a lovely relationship and they give him so much of the time.
He's very fond of Greta, the head nurse there, to the point that he actually cries when he has to go home.
I think Owen's case illustrates the value of the 100,000 Genome Project perfectly, namely that it has allowed a condition which is not at all easy to diagnose clinically to be identified, but also initiate treatment.
The 100,000 Genomes Project has laid the foundations for the new NHS Genomic Medicine Service to make sure that children like Owen have the best chance of getting a diagnosis as early in life as possible. That's right, Owen.
I just want Owen to have a full and fulfilled life. And as part of this project, we found out so much about Owen, and he has the best chance now.
Okay. I think that's a very powerful video, but I don't know what. So, I'm not going to show you any of the other ones, and I have told you about the CD16. You want to stay on this slide? Yeah. Sorry. I've already told you about the CD163 biomarker that we have in vasculitis, but I also know that things are moving on at pace within vasculitis, within our condition. I suppose to me, a naive. I'm not a scientist. I'm not, I'm not a doctor or anything like that. But, whenever I was diagnosed, I was told we don't know what causes it. And of course, being a young mother, your initial thing is it genetic? Is any of my children going to get it? And that is a real concern. And initially I was told, no, there's definitely no genetic thing in it at all. And now I know that it's something, it is a genetic predisposition, and it's something that triggers it within us. So I still think that it is possible that some of my children will develop something like this. But I suppose the advantage we have is that we know what it looks like. And if something starts going wrong, we'll be able to get it addressed really quickly rather than trying to put a jigsaw puzzle together. And with the work that ye are all doing, I thank you for the work that you're all doing. And that is changing lives. So I'm going to finish. Oh, no, I'm not going to finish. I'm going to ask you, did you find the talk useful? And would you incorporate anything? Have you learned anything, in this? Have you thought more about how can you get patients more involved in the research process? It's all right if you say no, but it's just, just open the mind a little bit and see, is there an opportunity? And the other thing is, don't be afraid, to ask your patient, and be transparent. Be transparent. I was talking about information leaflets earlier. We're trying to design information leaflets for our clinics. And the traditional drugs that are used in vasculitis are steroids, chemotherapy, and biological drugs. And one clinician said, "Oh, we can't say chemotherapy. We'll say azathioprine, and we'll say cyclophosphamide and whatever." And I said, "But you have to say that it's chemotherapy. It's not aggressive chemotherapy like what is used in cancers or that, but it is a kind of chemotherapy." And if this person, he says, "Oh, they'll all think that they have cancer." And I said, "But if you send them out and you tell them that this is the names of the drugs that you are on, or the pharmacist is going to say, this is the name of the drugs that you're on, they're going to find out they're on chemotherapy. And then they're going to think, oh, they never told me I had cancer." And you've created a whole scenario for nothing. Just say it out, and then if there's a question, you could say, "It's not aggressive. It's not as aggressive as if you had cancer. It's just a different regime, and it works." So, and these are some of the resources, if you were looking to try and get patients involved in research, in the research process. These are very good resources. There are many more, many more. So, and I'll thank you all very much for listening. And if you have any questions, I'll take them.
So, Julie, thank you very much for this very inspirational talk. And it's indeed, I am already, I have to speak for myself, struggling with all the computer stuff and things which are not working online. So I can imagine that for you it must be even worse. So thank you very much for having bravely just went out there and spoke to us. I enjoyed the talk a lot, and I think, as I can start off a bit, that for research grants these days, the European ones from the AGP, they do ask to involve patient organizations and actively in the design of the study, and it is also necessary for get funded. So I think it's more and more also from those parts acknowledge that it really is necessary to include patients. So would anybody else share their experience? Yeah, first you're getting there.
Just a quick question. I mean, obviously we're all here looking at
the genetic basis of diseases. And, you know, in my interactions with different patients, you get a very different response. I think, so, what is your experience with, is genetics a scary word? Is it something that puts people off or actually engages people more? I'm sure there's no single answer and each person has their individual response to it, but it would just be interesting to hear if you have any experience of heard, you know, in your case and in other situations, what's your response to genetic research? Because I always feel like that's scaring people off.
It is very scary. Any research for patient community can be very scary. But I think the big thing about genetics is that it's such a powerful tool. And there is a fear of that kind of information falling into the wrong hands. That's the big fear that is in the patient community, I think. And that's why I say, like whatever you're doing, be completely transparent, and and we have GDPR, and we have, there are restrictions. There are, patients' data is very safe, but it's tried to get the patients to understand how that works, and that it's imperative that it is kept safe and that it's not going to be abused. I think they have feared that a data breach or that it could be sold to insurance companies or things like that. But it's just, being able to reassure people that that kind of thing doesn't happen. And I think the other thing, in Ireland, there's a huge company, a big genetic company set up, but it's a private company, and there's a lot of suspicion about that, about why why are they getting paid money for this, or I'm not putting it across very well here, but it's just that it's seen as a money-making thing. It's not seen as in the public interest. So that's would be that.
Hi, I was going to ask, it's sort of a sensitive question, but often the diseases we're particularly interested in affects very small children. So it's usually the parents who often are engaged in public patient involvement because, a newborn baby or a 3-month-old can't be involved particularly. But it's how, or do you have any suggestions of getting, so some of those parents are fabulous, but they're very, very active, they're very online, they're very everywhere. And then as that child grows up and they get to be 7, 8, 12, 14, they no longer want either the level of engagement or their parents' level of engagement in terms of putting them on Facebook, YouTube, Twitter, whatever it is. Do you have any suggestions about how you handle that? Because some of these trials, particularly I'm thinking about gene therapy trials and things, they're 20-year trials. We reconsent them when they get to be 18. But it's a long time where you have a child who has very definite feelings and opinions and a parent who may not have the same ones in terms of all of that.
I suppose personally, I would think of things like play therapy and things like that, using that as a way of finding out from the children how they're actually feeling about these things, and maybe if the parents are present, while it's happening and that kind of thing. But I think it is a tricky one because the parents have guardianship and things like that, and they have to protect their child. But at the same time, the child is growing up and they're still not at that age that they can make that decision legally. But they have their own opinions, and I think it's just trying to listen as you go along. Incorporate age-appropriate activities that you're going to be able to find out that kind of information. That would be my suggestion. But also, Jenny Preston is fantastic at this kind of thing. So, okay. And she hasn't paid me to say that. It's all right.
So are there any further questions?
Thank you very much indeed for giving us that important perspective. I've been thinking about how to elicit what it is that patients and families want from research. And I was thinking about the James Lind Alliance process. I wondered if you had any experience of that you could share.
That's a really good experience. It's so thorough. Because everything is on the table at the very beginning. Absolutely everything. is on the table. And it's a very joint process. The project that I was involved in, it ended up that there were 10 solutions, and we had to get it down to 3. And it was, I mean, we were debating and discussing, and there was loads, but at the end of it, we had 3 solutions that everybody was happy with, because everybody had expressed their opinion. But I think it's an excellent process. So, yeah. Okay.
Okay. Well, thank you very much, Julie. And the next, no. The next, bye. So for the next part, I would like to introduce Marco Crimi from Kaleidos. Please explain us where it stands for as well. From Bergamo, Italy, and he will discuss a very interesting platform where also patients.
No, Martin is going to present. So for how long? It's 15 minutes. 15 minutes.
Okay, so in between and for now, Martin will present something for 15 minutes.
So good afternoon, everybody. Sorry, Marco, for the inconvenient. I will try to be pretty quick. First of all, thank you for having me today, because I'm very impressed by the work you are doing. I think that obviously genetics, genomics, this brings fear, but also a lot of hope. And I know a lot of patients who would dream to have a genetic diagnosis, and they wait for that. And even those where there is no chance at the moment to get any genetic diagnosis. So thank you for your work, and thank you for having me. So just a short introduction on IPOPI, because I have the privilege to chair this patient organization. This is a global one, and we represent patients with primary immunodeficiencies, genetic diseases most of all. And we advocate, so I'm a patient advocate for awareness, access to early diagnosis, and access to care. So this is all your field as well. And we are global. And we work along strategic planning where we want to improve access to early diagnosis and patient-centered care. Julie has given the definition on that. Build capacity and support national members, because we tend to have one patient organization per country. At the moment, we have 69. We want to educate, promote knowledge and data sharing, and last but not least, strengthen multi-stakeholder collaboration, and this is maybe why also I'm there today. So I have to say that Julie has said it all. So thank you, Julie. So I will be very quickly, I will take the patient perspective through this thing here. When we are not well, the first thing you have in mind is, what disease do I have, or my child, but also adults. Don't forget adults. And you need to have a name put on it. This is for sure. Then, is there a treatment? Of course. Where can I access it? And what does it consist in? And then, and you see you grow in the, that would be a Maslow pyramid. How will this condition develop? When you have a child with this condition, you think of him 10 or 20 years later. And for you, the same. How will I get old? Will the developing medical science be helpful for people like me? And then how can I support medical science? And then how can I bring my experience to this field? And then I would like, when it comes to genomics, genetics, and that stuff, to just put this in the patient journey. Before we have an odyssey, possibly, or at least symptoms, and then maybe we have this high throughput sequencing and get a diagnosis. And then there is an after. Just to say that what you deal with is just one step in a process that is a lifelong one. And this should be considered as a process itself that shortens the diagnosis delays, of course, but also ensures access to available treatment. So for this reason, there are many aspects that are at stake. Access to sequencing. Not every people in the world can access to that. Accurate diagnosis, personalized medicine, information, education, data, newborn screening, ethics, and policies. And for those, we have benefits and challenges. So I will just go through those different themes quickly still. Access to high throughput sequencing, this means availability, so facilities, expertise, experience. We often, as patient organization, get requests from countries from Africa, from Asia, to get some kind of diagnosis like this, because they don't have this in their countries. Affordabilities, have I to pay for that or not? And usability, for example, I had a case where a mother got a genome sequencing made in Israel, and she wanted to find someone who would be able to read it, to analyze it. Because people think that when this is done, and they have everything. So when suspecting a PID, but this can be other rare disease, of course, if appropriate, access to genetic tests should be possible for the physician and his or her patients. Accurate diagnosis and personalized medicine. I think that genetic testing plays an essential role in the diagnosis and the precision treatment of patients with phenotypic or suspected PIDs, and patients with various presentation of PIDs or other rare diseases should be tested using the platform that produces the best results. And I could see in the program that there are many options, at least several. Information and education, Julie has touched upon this subject already. Of course, this goes to physicians, and especially GPs and also pediatricians and internists for adults, but also of patients, because when will a genetic diagnosis make the difference? So the eligibility, not every patient needs to have this at first. Second, implication on diagnosis and on follow-up, and especially analysis. But also we have the question from adults. For example, an adult who has a CVID, common variable immunodeficiency, they say, well, I don't know my genetic diagnosis, but maybe they have found new genes, so should I repeat it? And how often should I repeat it? Every 5 years? What? So difficulties involved in management, interpreting, and communicating to patients the large amounts of data generated by the sequencing platforms, and patient organizations and medical societies could maybe join forces to more awareness as well as patient and medical knowledge. Data and registries. At IPOPI, we think that data are really very important, and registries as well, including for ultra-rare diseases. And of course, important of data collection, but here you know that. And patients, well, this is my thing, is patient self-reported outcomes, because I think that Julie has very well illustrated that patients have an experience that needs to be shared and needs to be taken into account. And I agree with her that human phenotype ontology may also be used by physicians, but also by patients, because this is also a common language to build, so to contribute to registries. In addition to that, of course, we need to ensure patients' data ownership, but also, and protection, of course, but also sustainable medical research, because sometimes, because we have too strict regulation on one end, we can't have research on the other one. So there is a balance to find between the two. Policies, genomics raises many social, legal, ethical issues that need to be addressed among multidisciplinary teams, including patient representatives. And so we need to have policies in place that ensure patients' access to the best practices, foster cooperation between researchers and clinicians to move forward, equity between the different countries, so that no one is left behind. Newborn screening, just at the topic, because we work a lot on that at IPOPI, especially to have equity within the European community. And of course, genomic may help newborn screening program identify specific genotypes that could shorten diagnosis odyssey. The implementation of high throughput sequencing would allow programs to screen newborns for a much wider range of conditions, including risks for later onset or chronic conditions, and more genomic information may increasingly help providers to refine potential treatments plans and to inform prognosis as severity of diagnosis or age of onset. And then, and this will be my last slide, Marco, the use of genomics in clinical settings have already highlighted also difficulties. Newborn screening program will have to address the long-term management of and access to vast amount of genetic data generated. They must balance suppression of certain results, moral obligation to disclose potentially valuable health information to families, and parents' rights regarding their children's genomic information. Also need to consider the potential harm of disclosing findings with uncertain or ambiguous implication for newborns that parents and their healthcare provider would find exceedingly difficult to interpret. So I wanted to have this in a very short thing, enhancing what Julie had said. Thank you for your attention.
Well, thank you, Martine. Are there any questions? Yeah, there's one question for you.
So it's not really a question, Martine. It's actually a comment, because all of this is, of course, the ideal situation as we would love it to be. But looking at the field of inborn errors of immunity or primary immunodeficiency, we are still, in most countries, a big end behind several other rare conditions, with lack of structured care, with lack of governmental support. And I think that is really very harmful for the patients, because what we would need is, when we do research, we would actually need a psychologist in the team. We would need someone to connect immediately with the patients when we are even thinking of, for instance, next generation sequencing and other kind of studies. Because of course, there is usually only research money involved, and that is one of the big problems as well. So when I look at for instance, colleagues with cystic fibrosis care, etc., that's where it goes like this. This is almost the ideal situation, with big backup financially, also from the funding organizations. And I think that's something that maybe as a community we have to strive to together, because that's how things would be much better for everybody. I think we would, as researchers and physicians, be much more able to offer all the support we need to offer, and the patient would feel supported rather than be I don't know, just an anonymous player. Yeah.
Yeah, I agree very much with you, Isabel. I think that, you know, us, and me especially, I am a strong believer in cooperation. I think that when joining forces, we can go ahead. I think that patients are more and more asked to be part of trials, not only because of ticking boxes in the requests, because this is also the case. We are committed in several trials at the moment because there was a case to tick, but also because I think that the mindset is evolving. When it comes to the different countries, I agree with you that the differences are really, really big. But when you begin somewhere, it improves. So when I look back, I am committed in the field since 1998, and when I see when I began, we had 30 PIDs, 30. So at the moment, we speak of more than 400. So yes, and it's about genes. So I'm really confident. Just let's begin and cooperate.
Well, thanks for these last nice words. And finally, Marco, you're welcome. So now it's time for Marco Crimi from Kaleidos, Bergamo in Italy, and he will explain a very nice platform which we could use as well.
Yes, my name is Marco Crimi, and I'm so pleased to be here today. So thank you, the organizer, for the invitation. I'm engaged for Kaleidos, that is a no-profit scientific research organization based in Italy, in the north of Italy. Our focus are on genetic diseases and mental health, and I planned to use this time, my slot, dividing in two parts. In the first one, I have some slides to present you, and in the second part, I would like to show you the platform I'm presenting here with the support of Dr. Croce. I'm a researcher as well, with my main interest on genetic diseases, rare diseases, and digital health. And I'm a genetic counselor as well. We have time to talk a little bit on genetic counseling in a couple of slides. Digital health is a very fast growing field. Digital health is a new discipline that connects projects on healthcare with digital technologies, with the aim to improve the healthcare services, improving the interoperability, because thanks to the digital system is much more easier for different facilities to be interconnected themselves, to facilitate the access to care, to guarantee the equity access for everybody, and to promote prevention measures thanks to the remote first aid with digital systems. And if there is in this time very fast growing interest on digital health, we have to thank you, this guy, because if the pandemic made a lot of disasters in different fields, we have to say that it played a crucial role to improve the transformation of healthcare in the digital way. And okay, too high voice. Can you hear me better now? Okay, thank you. So about 50% worldwide hospital now invested a lot of budget in the technology and in the information technology systems to improve their services, to fight against basically with COVID-19. And the projection for US market, we have to say that in 2022, about $30 billion will be raised in the digital health market. What happened in Europe, more or less the same. To date, we can count more than 600 companies that they are really active in the digital healthcare sector. And the European Commission have to consider important challenges. First of all, the aging population and the chronic diseases that absorb a lot of budget. There is also an unequal access and the quality of services, health services for patients, and we know pretty well what is meaning in the rare disease field. And there is a limitation availability of professionals in the clinical sectors. So the European Commission is investing a lot of billions of euros in the digital transformation for healthcare with the aim to facilitate the exchange of data in a secure protections environment, to promote the implementation of digital tools to facilitate the empowerment and the personalized treatments for everybody. Basically, the digital technologies in healthcare can be divided in 4 different areas. The most important one probably is telemedicine, so digital system that facilitate the connection, the relationship between clinicians and the patients. The second one is the wearable technologies, so the use of systems that we use every day to collect data for healthcare and to help clinicians to monitor the quality status of our life. Digital therapeutics is a very fast growing. Probably some of you already learn about Endovir. That is the first app cleared by FDA less than 1 year ago to improve the therapeutic approach for people affected, in this case for ADHD. And the last one is the EHR, the electronic health record, is very important. It's mandatory for any countries in Europe to have a database that can be standardized for collected for the collection in the most appropriate way of clinical records. What about genetics? Of course, even in the genetic medicine field is very important the use of digital applications, especially now that there is an important run to the consumer genetics, so it's very easy now to perform a genetic test. But we need the support of skilled professionals to help to support people who perform genetic tests, and it's not so easy to find skilled clinicians for genetic reasons. So some genetic counselors are professionals trained specifically to support people they have to perform a genetic test, and according with the National Society of Genetic Counselors, the genetic counseling process is a key process to help families and the probants to interpret their personal and familiar history, to be more knowledge about the risk of recurrence, the prevention measures, and research and social facilities available for their needs. So through the counseling process, the persons can be more informed about the right choices to be made for their genetic health. Genetic counselors are spread all around the world, but they are most present in America, in the north of Europe, and in Australia. And we have already learned from the previous slide what are the roles. However, it's important to stress that genetic counselors are very important to collect the most important informations by patients and by their relatives, and to return accurate educations, and to facilitate the relationship between the persons and the network of services for them. So our idea was to elaborate, to develop a platform that can be supportive for professionals who are involved in the genetic counseling process, and to help patients to be more acknowledged when they have to perform this kind of assistance. We started to develop this platform at the end of 2021, when Kaleidos, my company, was able to find the most appropriate funds to develop this system. We already submitted the intellectual property rights, and on about in December last year, there was the official release of this platform online. This is an award that the Italian Chamber of Commerce gave us. And some months ago, we were in Dubai, invited by the Ministry of Foreign Affairs to present this platform. So the goals, again, is to simplify the genetic counseling process through digitalization to facilitate the relationship between patients and healthcare providers and professionals, reducing the time needed for the in-person meetings, and to increase the patient awareness on genetic hereditary principles and other important things. Who are the target of this platform? Of course, professionals who work every day with the patients with genetic disorders, but also clinicians who they not assist in the clinical routine their patients, such as cardiologists, pediatricians, oncologists, that they care about patients with DNA mutations sometimes. But other target, of course, are patients who have already received a genetic report and they need the support from a specific expert. The platform is divided in 3 different areas, and in each area there are different software, different algorithms. The first area aims to educate the patients about the basis of genetics, and everything started to obtain the informed consent. What we provide to our partners is a software, is a technological infrastructure. So we don't use our informed consent. Our partner can apply, can upload within the platform is a standard informed consent, and their patients sign digitally their informed consent. We develop also a chatbot. That is the first chatbot specifically focused for assist genetic diseases. And there are within the platform some videos built with a very easy, simple language to educate patients about genetics. And so far there are 5 videos in 5 different areas of genetics. After the first module dedicated to educational, there's the second one that is for data analysis. So following the platform educate the patients, the platform started to collect data from the patients, and basically I mean data from his familiar and personal life. So there is a module to build the anamnesis and a specific module to build the genogram, the pedigree. Both these resources will be released to the medical doctors that can save in his computer both these tools. There is also a system to exchange files and information. So using this platform, the patients can't avoid to use inappropriate systems such as email, WhatsApp, or other socials to share clinical data or the clinical documentation with his physicians. Can use this system, and this system is able also to manage VCF files that are, as many of you already know, files from NGS platforms. And we are now in the process to develop bioinformatic tools to analyze VCF files to perform prioritizations of variants, as we learned yesterday. And so the first module dedicated to educational, the second one to analyze data, and the third one is the clinical feedback. So within this platform, the clinicians can can make a televisit with his patients and to release him the clinical report, the final clinical feedback. Quite fast to rebuild with you the operational flow is the physician who invite the patients to access the platform. The patients, without the invitation from the clinicians, she couldn't access to the platform. In the first stage, so it's the platform to inform patients about different things, and in the second phase is the platform that collect the data from the patients and to make them available to the clinicians. In any step, in any time, the clinicians, of course, can access in the right module to correct some information regarding the patients who that maybe are inserted in the most appropriate way. This is the bioinformatic tools. We are still working on this. And the clinician can also ask the support of another professionals. And all this process is built in the correct way to guarantee the privacy of the patients. The final step is the televisit and the clinical feedback reporting. Here is the list of the most important privacy certifications we obtained. The access to the platform is only made OTP, a one-time password, to support to make the most appropriate way for the accessibility. And all the platform, both in the front end and the back end, is built with cryptography. We already obtained a legal opinion by a third authority. And the patients, as always, the right to be cancelled according with GDPR regulation. This is just one slide for another platform we built about a couple of years ago in telemedicine, another platform for digital health. This is a telemedicine system developed with technology, the white label technologies, to guarantee our partners to use this technology as they built. It's 100% rebrandable and customizable. The future plans for us, of course, we are looking for, we are starving for a new partnership, patient advocacy organization, academia, universities, and other clinical facilities. We are planned to submit the certification for a medical device. The limitation to date is that all the platform is in Italian, but is already ready to be translated in English or in any other language, and we are searching for European funds. For the information technology development, the most important steps are to make interoperability with tools for diagnosis. We already know about the HPO terminology, and we are building the software for variant interpretation and, of course, prioritization. So thank you for your attention. These are my contacts and all the partners that we are collaborating with us.
Marco, thank you very much for the very inspiring talk, because indeed I think that's the way to go for in the digital time we are in. Would you like to have questions now, or first want to demonstrate?
As you prefer.
So, does anybody have any questions? So I think it's very important for the data sharing part, and indeed be encrypted and follow all the regulations. because we, as clinicians and researchers, all want to share and we want to do many things, but we're often not allowed to by our academic institution. So, so how do you envision to collaborate with other systems and other European countries?
Yes, absolutely. Would be mandatory for us. The first step, of course, is to translate the whole platform in the targeted language for our partners. But we are open to this kind of collaborations, of course.
And how does it relate to patient systems like Epic, like the platforms hospitals use? Could it be involved there as well?
Absolutely. The limitation, the challenge for collaborations with a public entity, at least in Italy, is for the informatic point of view. All the Italian hospital, but I think in other European countries, they are, the software in place are built with a specific language, informatic language, that is HL7 FHIR. So we are using this language for some specific tools within our platform, so there could be a 100% interoperability between our platform and the system in place for other hospitals.
Okay, very interesting. Other questions? Then I think it's time to
move to the second part, so I would like ask Stefania to join me. So, I never met Stefania before yesterday. Was the first time, so I asked her to, I invited her to register within our platform as a patient to make an experiment. We are scientists, we like to do it. Experiment. Okay, this is the landing page of our platform. Again, as everything in Italian. Okay, so there are two different profiles, one for the professionals and the other one for patients. Now Stefania was invited by me as a professional to access to the platform, and she already signed digitally the informed consent. I will show later when I enter with my professional account. And in the platform, there are 5 different videos available to the patients in different areas: recessive mutations, dominant mutations, pharmacogenetics, oncogenetics, and a non-invasive prenatal test for pregnant women. And you can see on the right the GeneBot. GeneBot is the chatbot specific for genetic diseases, as I mentioned before. So these tools helps patients to be more knowledge and to be more informed about the basis of genetics and avoid the first period during the genetic counseling to make the same questions to the medical doctors, because patients is already informed with these tools. And in the second tool, there is the anamnesis. So is a pre-filled tools with a lot of different questions and selections for the patients. Is a tools built specifically for patients with genetic disease inherited. And it's quite long because, you know, there are several informations that the professionals, the physicians need to know. And there is also the genogram. So Stefania already built, using this automated system, the genogram that can be also more complicated according with the familial history of the patients. And can be signed people with genetic disorders, if they are already alive or not, and all the informations needed for a genetic tree. And in this tool, you can see the my name. I was the medical doctor to invite her and to access to the televisit modules. Okay. There is also the possibility to share documentation with the physicians, including also bioinformatic files. And the patients can find here the clinical feedback from his doctors when he's available. Okay? So I alto, se no faccio vedere. Now I log out and log in again with my account. This is Stefania, my patient. I can see that she already signed digitally the informed consent. The platform is composed by different modules, but of course it's not needed to use all the different tools. For example, with some partners, they need just a system to collect the informed consent digitally from by remote from their patients. And I can download the signed consent that is signed in a correct system. That is legally appropriate. Okay, and I can receive here all the genogram, the anamnesis, the documentation that Stefania will share with me. I don't know if you have any other question.
It seems rather clear and simple to use. Are there any questions? So what kind of data can there be in? So the genetic data can be in, you can see the counseling and stuff.
Yes, we can receive data on the familial history, as I showed before with the anamnesis system, with the pedigree, and the system can manage any kind of PDF or Word with clinical relations or documentations, and bioinformatic tools, bioinformatic data as well.
Okay, so if I may ask, Julian Martine, would you be able to, would you use systems like it, bioinformatic systems, to talk to your medical doctor? Would you see something missing or?
I think it would be a very useful tool, especially, you know, like for people that are rural, living in a rural place, they're not living near their hospital or that, and it would be a really effective way of being able to communicate with their clinicians, I think.
Thank you. Yes, the goal is not to replace the professionals of people with expertise in genetics, but to support them in the process of assist their patients. And here we will also developing the system according with the HPO system to help for digital diagnosis using clinical terms of their patients.
Okay. Are there any further questions? I think Yes.
Thank you. I think as well that this is a very interesting tool. I would like to ask you, how do you see the way to convince patients, but also physicians, to use it?
Usually, genetic counseling process is really time-consuming, more than 1 hour and a half before starting with a study of the case and to answer to all the questions by the patients and to report the clinical feedbacks. So the goals here is to reduce this long time, and if the platform in genetic counseling, digital platform in genetic counseling can enter in the clinical routine, really can save a lot of time and help patients to be more knowledge.
Because from the patient perspective, sometimes the patient complains that the physician have no time. So you see, just keep aware of the fact that the announcement as first, this is very important to have a physical exchange. And then after that, this tool would be very useful, I think, yes.
Yeah.
Thank you.
Thank you. So, yeah, probably there are possibilities to use a system as this in the first phase during the triage of new patients, or in the follow-up after the first meeting, as you mentioned.
Yeah, thank you. This was really interesting. I was wondering if any data is required for the families of the patients, for example. Then would they also be invited separately to fill this information, or do you expect like the patient to fill all the data about the family history and other information?
Yes, the medical doctors have always the possibility to correct and to report new information if he knows that there is something lacking during the data insert data by the patients. And of course, any patient has the responsibility to inform his families during this process, as in the normal genetic counseling process in persons.
Yeah, related to this question, do you think that also the relatives of the patient, of the proband, also need to give informed consent in this case, because you are, of course, recording their data digitally instead of in a consultation? Do you need informed consent from all of the family members in this case, because you are recording their data digitally instead of during a physical consultation?
As in the standard consultation in clinics, so the patients, the consultant have to inform his relatives about the information he's releasing. So it's the same in a digital system.
Any further questions? If not, I would like to thank Marco for this nice presentation, and Stephanie for assisting.
So we're closing this 2 days of workshop. First of all, so thank you for the people in the room to attend up to the end. We hope that you enjoyed these 2 days. Many things. So do not forget to fill the form, and you will have 12. It's 12 points for the credit the accreditation. So that's 12 points. So do not forget to fill the form. Send them to Joe. For the EGP members also, you have the thing. Some days this presentation will be on the platform of the ESID website, because a number of people were asking for it. Not now, in few months. And so you will have access to the talk and the presentation. And we hope you enjoyed. And that's it. Marianne.
Yeah, thank you very much for everybody from the ESID, ERN RITA, and the AGP members as well. Thanks a lot for joining.